Okay, I think we're going to start. So it is my very great pleasure to open the symposium today and to warmly welcome you all here at UCL. Today's symposium is a partnership between UCL and the MSA Trust. And I went onto the MSA Trust website a few days ago, and they have a really bold vision. Their vision is to end MSA. And for those of us, and certainly for the 3,000 to 4,000 patients in the UK with multiple system atrophy, this is a bold but absolutely critical vision. As we all know, for those people who treat and diagnose this condition, it's very overwhelming for patients, for their carers, but also for clinicians who join them on this journey and feel often, certainly from my perspective, helpless in helping them both with symptoms and with changing the outcome. Patients will often say they just hope for a better day, a good day, a day where their symptoms are better controlled. And with that in mind, we should remember that everything we do here, from improving understanding to improving diagnostics, biomarkers, symptomatic therapies, as well as disease-modifying therapies, each one of those in themselves helps to make patients' lives better. And today is about bringing hope to that reality. The core values of the MSA Trust also sit on their website, and many of those I think we will try to invoke here today. Patients at the heart of everything that we try to do, and thank them for contributing to the research that will be shown here. Being open and collaborative is the only real way forward for us to help find a cure. To be respectful of each other, of course, but most of all for you as a community to be committed to helping further research and to stay involved. And I hope today we will see all of that through knowledge exchange between you all, through networking, and through really pushing the speakers on whether they really are making breakthroughs and where they think the future of research lies. So with that, I want to thank Valeria for putting together this phenomenal program. I'm never able to say no to Valeria, and I think that's a huge talent for anybody who has emerged as a leader in this field, bringing together the community, creating the infrastructure for research, and also leading a lot of that research. So thank you, Valeria. We're very appreciative of this. And thank you to the MSA Trust, of course, for bringing us all together. So I have a few specific things I need to say to you as well. The schedule is very packed, so please return promptly from your coffee breaks and lunch breaks and networking opportunities. We will run the talks back to back, and then there will be the opportunity for questions and answers in the 15 minutes at the end of the session, so please wait for that time. Food and drink are strictly prohibited in the lecture theatre. I say standing next to a beaker of water. And lunch is held off-site at the Bedford Hotel, and all of us will help direct you. It's just across the square. There are no fire alarm testing today, so if you do hear an alarm, please follow the route out to the fire exits, which are all marked. Okay, so with that, I'm going to now open the first session of the day today. New insights into alpha-synuclein genetics and biomarkers. So it is, of course, a huge privilege to introduce Sir John Hardy. John normally introduces me to most of the talks I give here at UCL, and he always describes me as a rising star. So I can very clearly say that this is a risen star a long time ago. And really, John needs no introduction. But of course, his contributions through understanding genetics in Alzheimer's disease and the discovery of the mutation in the APP gene has really transformed our understanding of Alzheimer's. He's made many seminal discoveries using genetics in many of the neurodegenerative diseases, including Parkinson's, motor neuron disease, and many more. He's had a large number of awards. I'm not going to list them all, although I'd like to embarrass him, and of course has received a knighthood for his services. I'm very excited to hear what he has to say about the genetics of synucleinopathies. So welcome, John. Thank you.
That was very kind, Sonia. I would describe myself as a sinking star, I think, actually. So why do we want to understand the genetics? Well, I mean, just to start by saying, I think that what we're seeing at the moment, finally, is that we're starting to see mechanistic therapies based upon genes that are being discovered. So I think that understanding the genetic bases of diseases is turning out to be a key part in developing treatments. Now, as I'm afraid I'm going to show, and I don't want everyone to get too excited, MSA genetics has not yet really taken off. We have not so far made any discoveries, though if people want to slip out and get a quick coffee now, maybe they should. So where are we up to? Well, I will say that when we found the synuclein triplication, Dennis Dixon looked at the pathology of the synuclein triplication, and this has actually now been replicated in two or three other cases than after the one that he looked at. We did in fact see MSA pathology in those cases. So if you really drive synuclein expression hard, you do get oligodendral dendroglial cytoplasmic inclusions. So it is there. If you push the pathology very hard, you get them. So that I think is a very important point. But you are pushing the pathology very hard. We decided to do a whole series. We really emboldened by that. When I came here, Henry and I started with others to look at to try and do GWAS for MSA, to do a genome-wide association study. At that time, we had rather few brains in the brain bank, and we published this paper, which was eminently plausible that there was a GWAS hit for MSA at the synuclein locus, and almost immediately thereafter, Ammar El-Chalabi published the same finding. Unfortunately, these findings are wrong, and but they're wrong for a slightly interesting reason, and that is that within the sample set, we had a few Asian samples, and at that time we had not corrected for population stratification. And the genetics at the synuclein locus are subtly different in Asian populations than they are from European populations. That might, of course, be important. And in fact, what we were seeing was an effect of population stratification in the data set. So the data is not reliable. How come it was replicated? Well, it was replicated because, in fact, Ammar El-Chalabi had the same samples as we had. There's the auditing, sample auditing. This is a continuing problem in genetic studies. Sample auditing was not very good. We didn't get any samples from Ammar, but he had samples which he had the blood samples and we had the brain samples, but they were the same essential people. So that's why. So this replicated finding is in fact unreliable. And actually, there have been some GWAS now published for MSA, and I'll just say that so far the synuclein locus has not come out in any of the GWAS. Whether it does or not as we increase numbers is an open question. GBA. Of course, GBA is really strongly associated with Parkinson's disease, and it's completely reasonable to expect GBA to be associated with multiple system atrophy. We've published from here on the brain bank, and you can see the very clear title, as is the second one down, and the Mayo group did the same thing, and they were quite clear that they didn't see any real evidence for an effect of GBA variants. In this larger study put together more recently, there is a weak association with MSA. One always worries when one sees a weak association of of a rare disease when there is a very strong association with a related common disease, because it only requires a little bit of what one might call diagnostic seeping to influence the results you get. In other words, if you've got a slight contamination of your MSA group by Parkinson groups, you're going to see an association. So, one has to be a bit careful. A similar example is that when we look at ApoE in frontotemporal dementia, we see an ApoE effect in FTD, but when we look very carefully at pathologically confirmed FTD, we don't see that association. So one has to be worried about seepage. This paper was also got a lot, well, you can see it's a New England Journal paper. This raised hopes that this was a finding in the coenzyme Q gene, a variant in the coenzyme Q gene, which was predisposing to MSA. This turned out, in fact, to be an Asian, this is published from Japan, this turned out to be an Asian variant. And what you do, it's changing because the databases are getting better, you look for a variant and then you look in the databases of that are available online of genetic variants. This variant was not in there because nearly all, at that time, it's getting better as I say, nearly all the variants were European variants. And so when they had looked for this variant, they had not seen it in what were essentially European databases. So this also turned out to be a contamination of, well, contamination is perhaps the wrong word, flummoxed, if you like, by the background, the genetic background of the cases versus the genetic background of the controls they were assessing. So again, a bit of a disappointment. Here we have the first GWAS. I'll just say straight away, synuclein is not on there, GBA is not on there. They report two variants in two genes called ZIC1 and ZIC4. The red line is for genome-wide significance, and you can see that none of the SNPs are genome-wide significant. So then this is not statistically significant. And to be frank, I think it's probably a false, well, not even a false positive, because it actually technically is not positive. But one has to, I would not work on ZIC1 if I was a cell biologist, yet anyway. So it requires confirmation in a larger data set. And that confirmation we're trying to put together. Henry and Viorica are putting that together at the moment. So why not more progress in case control? Given that, we always in MSA compare ourselves to PSP. Well, the numbers have been too small, I think, or the numbers of cases and controls have been too small. That's one thing. I think really it requires path confirmation. I don't think we should be doing, given the contamination of MSA clinical samples with dementia with Lewy bodies, Parkinson's disease and PSP, even though the clinical separations are reasonably good, as I said earlier, if you have some contamination, you're going to see signals relating to the contaminant disease. So I think we have to stick to path-confirmed samples. And it's a rare disease, and the number of samples are going up. They're going up, but slowly. The other thing I'll just say, and this is really a bit pathetic, but if you look at point 4 there, point 4 is what I think also was a problem. Henry and I were collecting samples, and we were funded to do other projects. So what we did is every time we had a few chips left, what for other diseases we save them for the MSA, for the MSA study. And the trouble with that is it makes putting the data together afterwards, because each one has been, if you like, assessed on a different chip. Putting the data together afterwards is very, very messy and difficult. So I know that's a bit pathetic, but it's true. The other thing is, I think that it's genetically heterogeneous. You know, we know that there is a slight shading of the pathology and the clinical symptoms between Europeans and Asians. To me, that indicates that there is some genetic background, and we should be separating them and analysing them separately from each other. I mean, in general, the Europeans have a Parkinsonian phenotype, and the Asians have a cerebellar phenotype. That's separating black and white when, in fact, of course, everybody is shades of grey. But the fact that you do get this type of shading of the clinical symptoms is an indication that perhaps there's something genetic going on. And as I'll show in a couple of slides, with PSP, separating the PSP clinical syndromes into different subtypes has turned out to be extremely profitable for Hugh Morris's group, and I'll show that data in a couple of slides. Christos, who's here in the audience, has taken a very, I'm going to say brave approach, because this is a very difficult thing to do. But we now know from the work of many, but I think most famously immediately by Prusner, that MSA behaves in, like, I don't want to say a prion disease, but it's a disease where you can spread the disease from one cell to another. And if you're in that situation, then of course one only needs to think of the somatic mutation in a single cell to initiate the disease process. And one can imagine a process by which, let's say, a single cell has a somatic mutation. Let's say, well, actually, as Christos says in the headline here, if you have a gene duplication in a single cell which leads to pathology in a single cell, then that pathology can spread throughout the brain, but in principle. I'll just add to that, of course, that one might expect under those circumstances that cell then dies. So having caused the problem, that cell then dies. And that's why I think this is brave of Christos to do it, because the murderer might commit suicide immediately after he's started the murder, so to speak. So this is really, and it's pushing the technology. It's really, but he's had some successes. So let's see. I think this is I have to say, yeah, it's a brave. I don't mean brave like as in Yes Minister. That's a brave decision. But I will just say it's definitely pushing the edges of the technology. Lessons from other diseases. Well, of course, I think I'm going to talk about PSP. This is what I started to say about the different forms of the disease. By comparing PSP Parkinson with PSP Richardson, I can never remember which way round, Hugh's group, Ed Jabari in Hugh's group, really showed this very nice finding that genetic variability in a locus called the TRIM11 locus was responsible for that variability. This, we now know, makes etiologic sense because the TRIMs are part of the ubiquitin proteasome system, and the TRIMs are involved, and this seems to be involved in tau degradation. So what we're seeing here is a gene which is very likely involved in tau degradation, influencing the phenotype of PSP. I think this is a completely plausible gene, way over genome-wide significant. And just by the way, we've just done a GWAS of Pick's disease, which is 3-repeat tau, and found a gene called TRIM25 is involved in Pick's disease. So I think another ubiquitin proteasome. So I think this is plausible and is pointing at the degradation of tau as being important in the phenotype of PSP and, for that matter, Pick's disease. And then looking, I don't know what's happened to that slide, looking at the other, all of the GWAS that I've shown for MSA have been GWAS of case control. What we have been doing, again, it's led by Hugh, is looking at the rate of progression of PSP. And again, this has given very plausible and interesting findings. And the finding here is LRRK2. So LRRK2, which of course is a famous Parkinson gene, LRRK2 influences the rate of progression of PSP. And I don't know how many of you realize, but quite a high proportion of patients with LRRK2 mutations actually have tangled pathology. So again, it's plausible. But it's also showing that what we need to be doing is collecting longitudinal data. In this is a diagnosis to death, was the phenotype, and doing a genetic analysis of that. And that's clearly something that we need to be doing for MSA. So what is needed? Well, better sharing, I think. I think that we need better sharing. We need to be able to more autopsies. And I know that Henry, you said that no one could say no to Viorica. Viorica has been persuading people, and Henry has written to every brain bank in the known world, getting samples too. So we're doing our best. Viorica and Henry are doing their best to increase sample size, to add on to the GWAS that I showed from Gunther Huglinger. And I think that we can say that we're going to have a GWAS of something like twice the size of that last GWAS. And hopefully that'll start to get data. And I know, of course, that they are being very careful when they can to get longitudinal data, which I think is going to be crucial. Another thing which I put, which is kind of boring, but very important, given what I've said, is that we need to have sample audits. One of the problems, I'm all in favour of patient privacy, but I have to say that that the way that samples are recoded every time they change hands is an absolute nightmare. And it's really hampering research. I mean, I'm not advocating we obviously have to respect patient privacy, but it really is a problem to trace a sample back when it's been through 2 or 3 hands to understand which sample it is. And then quite a lot of centres don't allow sharing of genetic data, raw genetic data. And that means that, geneticists can always tell if you have had the same sample because they just compare the genotypes. But if they never are allowed to see the raw genetic data, then sample auditing becomes a real problem. And, people are, of course, in all the brain banks, and clinicians are all very busy, so getting information from them, who was that patient, is actually difficult. They don't always, I know that a lot of us in the lab spend a lot of our time chasing samples back to their source. So that's where we're up to. I think, whoops. So I think that we're very likely to make real findings in the next 2 or 3 years, actually. There's 2 manuscripts in press, which I have seen. I will just say that I have seen both of them now, and I believe one of them. So that's where we're up to. I think we're going to start to see real findings. So I think that's where we're up to. So the future's better, and we're getting funding now also from the ASAP group, indirectly from the Fox Foundation. They are starting to fund rare Parkinsonisms, including MSA research. So I think the funding situation is going to get better for this sort of work as well. Thanks very much.
Okay. Not to relax too much, John, because there'll be questions, I'm sure. Thank you so much, John. So now let me welcome Professor Claudio Soto, who I've chaired a session with him before, but brilliant to meet in person. So thank you for coming. He's a professor of neurology and director of the George and Cynthia Mitchell Center for Alzheimer's Disease and Related Brain Disorders at the University of Texas Medical School. And I had not known, but Claudio originally did his PhD at the University of Chile in 1993. And really has spent the last 15 years understanding protein misfolding and has really rocked the alpha-synuclein world, of course, in the last few years with the seed amplification assay, which we think is going to really be transformative to diagnosis, but also to understanding. So very, very pleased to welcome Claudio here to give us a talk.
Many thanks for the introduction. I don't know which microphone. Glad to be here. Thank you, Virica, for inviting me. I wanted to come last year, but my baby was about to be born, so I couldn't come. I'd like to talk to you today about the advances with the seed amplification assay. I'm going to go very briefly to the background and show you some new results that we have recently. So let's see. First, I need to start with the conflict of interest. I founded the company to develop seed amplification assays for various diseases, and as many of you know, the Synuclein test is being commercially available in the US. So I have to share that piece of conflict of interest. This is what we think is a disease pathway in MSA, which is really an adaptation of any neurodegenerative disease, right? So where we think that everything starts with the misfolding of a protein, in this case the alpha-synuclein, form these oligomeric structures that may be toxic to cells in the brain. This aggregation process continue going to form these fibrillar aggregates that form this deposit that you see in the brain. In the case of MSA, of course, the deposits are called glial cytoplasmic inclusions, GCIs, right, in oligodendrocytes. Any of these events, not necessarily the final accumulation, but maybe the earlier intermediates, will cause neurodegeneration, cell death, and cell dysfunction, and then that will lead to brain malfunction that finally lead to the disease. What we know from this is that not only in MSA, but in many other diseases, the same pathway, of course, different proteins, different intermediates, different type of aggregates, but similar pathway, that the clinical symptoms come very late in the disease. So the process of protein misfolding and aggregation start years, if not decades, before the final outcome. So that gives us an edge in terms of early diagnosis. If we can detect some of these earlier processes, then we may be able to predict who may develop the disease 10, 20, 30 years later. So that, of course, is essential for, as the Alzheimer's field has shown us, the earlier you go, the more benefit you can get in treatments. And I think the MSA Trust goal of eliminating MSA, it's a great goal, and I think it's achievable if you can couple an early detection system with a good safe therapy you can actually slow down the process enough so that the disease never show up. That's what we really would like to do. So that's what I want to show you. One piece of this, we're also working on treatment, but I'm going to show you the piece of the detection, which we have been more developed. So, we all know that the synucleinopathies don't come in one flavor. There is different diseases that are associated to alpha-synuclein, PD, DLB, MSA, and the prodromal versions. We know that, and we think that different diseases are associated to different structures of the aggregates that are formed, and then they localize in different cells and in different areas of the brain. So what is our strategy for sensitive detection? So we know from, again, many other diseases, and what I'm going to talk to you today is more about MSA, but we have applied a similar concept to seed amplified various different type of aggregates. The work actually started with prions over 20 years ago, and now we are detecting, basically using in clinic the technology to detect. And then we moved to the others, amyloid beta, tau, synuclein, etc. But the whole, what is behind is that we think that the process of protein misfolding and aggregation follows this seeding nucleation process in which everything starts slow with the early misfolding event to form this misfolded intermediates that then start to do protein-protein interaction to become stable. If they don't interact with anything, they're very unstable and eliminated. But if they start to, form oligomers, they become at some point stable enough so that they can keep propagating. And these oligomeric versions or early aggregates, they have this unique property of what we call seeding, that they're able to recruit more of the protein and make these aggregates to grow, and then the process become more exponential. If you do this in a simple experiment in the lab, you follow aggregation versus time, you will see this typical curve. If you add a seed, preformed seed, this silent phase we call lag phase, it's accelerated and the whole process becomes accelerated. And so that's what our rationale. If we take a patient sample, let's say CSF, blood, or whatever, and if patient samples have seeds, we add the substrate for the growing, and then we will be able to detect aggregation if there are seeds. If the sample doesn't have seeds, then you will not see anything. It will take a long time to see aggregation. And that's basically the principle behind the seed amplification assay. One trick that we did, and this is again coming from what we did in the prion field, to accelerate this process, we do a PCR-like process in which we do cycles of amplification, because what happens is that if you, let's say, put oligomeric seed, and the patient samples, especially CSF blood, would probably have very few seeds, right, that you cannot detect by any other technique. So if you take this very small amount, in this cartoon I have one, right? If you have one seed, one aggregate, it will grow, and it grow in a bidirectional way, but it will take a long time for the process to be complete. So what we do is we fragment, after some time, we fragment this aggregate to release more units that can each of them seed further aggregation. So, and then when they're cut it, let's say fragmented, then each of them keep growing at the expense of the normal protein, and then when they grow, we can fragment them again. So this is a cyclical process that we have been using as a trick to do this in a few hours in the lab instead of taking the years that takes in the brain, right? With fragmentation, we use different sources of. It can be sonication, it can be shaking. So the seed amplification assays were previously known as PMCA or RT-QuIC. This is what we use in the prion field. So several of us working on this, came together and then say, okay, well, let's adapt a new nomenclature for the new assays. So just to differentiate from because the previous assays are replicating infectious material, infectious prions. So we didn't want to have this connotation, again, a difference between PMCA or RT-quick and all that. So we try to unify everything in this new concept of seed amplification assay, which I think describes better what we are actually doing. So just a nomenclature thing. I don't have time to tell you, but we have published a number of articles. I want to touch a little bit on the Nature paper, but the others, just to show you that the technology has been extensively used, mostly for Parkinson, and mostly in CSF, although we have now assays working in skin, and we are advancing very fast into the blood assays. But most of the work so far has been done in CSF. We published a number of papers. Some of these articles are actually done with different groups, where the same samples were tested in different groups, and we got impressively similar results. The last one is this Lancet Neurology that we published last year, where we show in a very large cohort of PPMI patients a very high levels of sensitivity and specificity. There are several more papers coming out from this study that will be reaching probably this year, the literature. But it's an exciting time. So let me tell you a little bit now, go back to MSA. So this was a chance. I mean, we didn't really think of this, but it happened when we start testing some more MSA samples, because initially we were just working with PD, we didn't see anything. But when we started testing MSA samples, we start seeing that the product that we are getting is different in MSA than in PD. These are, for example, here in the bottom, there are samples of patients. Each line represents a different patient, and these are the 94 patients with Parkinson's disease. You can see that the majority of the lines have a similar pattern that gets to a fluorescent reading of 5,000 in this particular machine. The number is very different in different machines. But what it matters is the pattern. Maybe I will make a parenthesis to say that what we use to measure the aggregation, it's a dye that is commonly used in the field to measure formation of amyloid, thioflavin T. And this thioflavin T, what it does is a fluorescent molecule that, when it's not bound, it's not fluorescence because it's moving. So it gets bound, intercalated into the aggregate, it gets immobilized, and then it doesn't move anymore and gives fluorescence. So you can imagine that if you have different structure in which the thioflavin will be immobilized in a different way or bound in a different way, you will get more or lower fluorescence. And that's precisely what we saw in the case of MSA, that when we analyze the samples, you see all follow this blue pattern in which the majority go to a lower fluorescent reading, maximum fluorescent reading. Of course, there are some few that looks like Parkinson. I think they're misdiagnosis. And the other case also, there are some few that Parkinson looks like MSA. And what was interesting is that we can easily differentiate by just looking at the readings whether the sample has one origin or the other. We got a very high sensitivity and specificity to differentiate PD from MSA. This was what we published in the Nature paper. We have been doing many more studies and increasing the number of samples, and we kept the good levels of sensitivity and specificity. I know some groups working with seed amplification assays say that they cannot detect MSA, and that's only because the conditions they use. The fluorescent reading is very low, so you have to use a good set of buffers, pH and everything, so the thioflavin can give good fluorescence at that condition. If you don't do that, then you will see no fluorescence in the MSA, and you think it's negative. It's just, when you see SAA assays, you have to be looking at what really protocol they use. And there are various different protocols in the literature. So we have consistently getting this also in the brain samples, and it's very clear. So what is interesting is that even though the fluorescence is very different, the amount of aggregates is the same. When you look at the aggregates by other techniques, you see the same type of reading. So, but again, we have a good differentiation between PD and MSA. One thing I just very quickly go into that most of the previous studies were done with the our original SAA assay that takes several days, maybe a couple of weeks to run. we more recently, in collaboration with the company Imperial, we developed a new assay we call enhanced assay that it takes only 24 hours and it still can differentiate PD from MSA. So many of the studies that we are doing now, we're using this faster assay. And this is just to show you the performance. So these are CSF samples. You have the PD and MSA. Look at the different scale. Well, it's the same scale, but it's cut. So the PD goes to, in this machine, 200,000, I think, whereas the MSA stay in, it's difficult for me to see, in 5,000 or so. So it's a clear black and white reading. So that's CSF. We also do it in brain. In brain, you have to dilute it. If you put the brain directly, brain homogenate, it will interfere with the assay. But a dilution of 1,000, 10 to the 4 and beyond, you can use the brain as a material to read. And you can actually dilute the brain to 1 billion-fold, and you still see the signal. And again, you keep the difference between the PD and MSA. So we can do that. We can detect specifically PD and MSA. In the Nature paper, we show that there are several differences that we can differentiate the two, what we call conformational strains. What I would like to spend the few last minutes, because this is important for research, not necessarily for diagnosis, but the question is, does seed amplification maintain the properties, biochemical, biophysical, structural, biological properties of the original aggregates present in the brain? So if that's the case, then you can use it for research, you can use it for drug screening and things like that. So let me tell you some results we have in this. So one of the things we did was to check various dyes for interaction, differential interaction between the PD and MSA. So this is the one in the top that we reported in the Nature paper. You can see in the red, you can see very well the signal with PD, and you don't see much with MSA, even though both samples have the same amount of aggregate. This is at the end of the reaction. Yeah, it's a reaction. We screened many other dyes, and we found one that only read MSA and doesn't read the PD. So we have now dyes that read only one or the other, so you can actually put, if the dyes, fortunate enough, they have different colors, you can actually put both, and then if you get the blue signal, you get one disease. If you get red, you have the different disease. So that's what we're going to in the future. But importantly, this dye that we use for MSA was good to do histology, to look at in histological samples, and we take slides from PD and MSA, and you can see that even though the PD has Lewy bodies, it's completely, the stain with this dye is completely black. Whereas when we have GCIs, you see the staining. So what this indicates is that what this dye see in the brain is the same that it sees after amplification. So, it goes in the same relationship. So that means that probably some of the properties at least kept. The other thing that we did was to do denaturation assays to see how much stable these aggregates are after amplification or before amplification. So we took the brain, non-amplified, and we subjected to different concentrations of guanidinium. And you can see the pattern that the PD is less stable than MSA. And the same thing we see with the after amplification from the brain or after amplification from the CSF, suggesting that, again, the aggregates retain some of these properties. The other thing was to do going more into biology. We use this material in the cell reporter assay that Mark Diamonds and others had been working on. So this is a synuclein contract with GFP that gives fluorescence when it's aggregated. So as reported before, the MSA, it's so much more efficient. That's why Prusiner, mostly from in vivo experiment, called that MSA is a prion and PD perhaps not a prion, but I think it's just a matter of quantity and a matter of time. So the PD, you can see something, but much less. The most important thing is that the morphology of the aggregates when you start with MSA or PD is different. So you can see, more time here is more clear. You can see how these fibrillar structures in MSA, whereas the more punctuated structures with PD. What is interesting is that when we give the cells brain without amplification, we get this pattern. But when we give the material after amplification, we again get the same pattern, suggesting that these biological properties remain. So the last thing I want to show you, well, before some of the structural work, it's that we also did the in vivo experiment using this transgenic mice. And this is just the characterization on an inoculum to show you the LVD versus MSA, and again, this the difference in the seed amplification assay signal. So when we put this, let's see if I can. When we put these samples into the animals, only the MSA, as reported before, verprusinate produced the disease. And you think the disease is very clear. I mean, the animals barely move at the end of the disease and they die. So they die early on. And then when you see the pathology is only present in the MSA and not in the PD. We think this is just a matter of quantity. We are trying to overload now with the PD to see if we can see a PD. This is brain homogenate. It's all brain homogenate. We can induce the disease by PD brain homogenate. But because of that, we were only able so far to do the comparison on MSA. So what we did was to inject the MSA brain homogenate from a patient and inject the MSA amplified from the CSF at 2 different concentrations. You can see here that when we use a high concentration, the disease is very fast, around 100 days, and very much synchronized. This is precisely what we see in the prion field when we inject prions in the animals. When we put less concentrated, 100 times less, you see, and this actually happen to be similar concentration of what we have in the brain. You can see that the disease comes later and at the similar time of the disease that comes from when we injected the brain. So in terms of survival time, it's very similar. Also, in terms of pathology, you can see how the MSA brain and MSA amplified, they're very similar in the places where they induce pathology. Also, the presence of protease-resistant aggregates is very similar. We went a little bit more in deep, and then here we just characterize pathology in different brain regions, and you can see the same type of pattern. Each number represents a different brain region, the amplified product and in the brain. We also look at the morphology of the aggregates. In the case of, in this case, the majority comes as what we call ring-like, that has some reminiscence of what you see in the brain of MSA patients, and whereas some few comes like a Lewy body type. And then we look at the ring-like versus Lewy body type. Again, we produce the same type of results with brain or with amplified material. The properties of the product is also the same. This is coming from the brain. I'm going to move on. This paper is probably coming soon, I hope. The last thing I want to show you is that we can actually look at the atomic resolution structure, the revolution that cryo-EM has produced. What we did was to take the amplified product from CSF and do the cryo-EM studies and finally got to a structure that has a resolution of around 3.5 Armstrong. What was interesting is that when we compare this structure that we get from the amplified product with the one that was published by the Chairs and Goddard group, we get very similar structures with this value of comparison around between 1.7 and 2.3, depending on which of the protofilaments you compare. So at the protofilament levels, they are very similar. They pack a little bit different, and that may have to do with different environment in the brain versus in the test tube that we use, but the structure of the protofilaments is very similar. We got the same actually for tau, and we're not going to have time to show here, but again, we can also amplify the tau structure. This is comparison our structure in blue with the PD or DLV structure that the people here in the UK publish, and the RSMD value is very high, so meaning that they're not similar at all. So to conclude, I would like to say that we have this high sensitive and specific detection of alpha-synuclein aggregates in biological fluids using our SAA assay may represent a great opportunity for objective biochemical diagnosis of this disease. We can differentiate different forms of Parkinsonism, and we can differentiate conformational strains in patients affected by PD or MSA. And the properties of the product that we produce, it's very similar to the one that is present in the brain, suggesting that the seed amplification faithfully retain the properties of the seeds. I think the SDA technology might be used to advance also the new biological definition of these diseases that, is being very hot these days. You probably have your own opinion, each of you. But it's an interesting possibility that will allow faster and better clinical trials. This is just the people in the lab trying to do misfolded versions. And here are the list of people. I really don't have time to list all of them. The one in red are the ones that contribute the most to this work, and collaborators all over the world. In the future, we hope to list the people here as collaborators, because we really depend on people who can provide us good samples. And of course, the funding agencies for support, mostly for this work, the Michael J. Fox Foundation, as well as NIH. Thank you so much.
Thanks, Claudio. Very illuminating lectures, maybe less so on genetics than. Okay, questions. Any questions? Henry.
I don't need the mic because I'm quite close. I don't need to ask John anything because I usually ask him in the coffee shop. So I have all his secrets. But Claudio, I mean, very interesting about this for diagnostics. It's something that we're doing here at Queen Square, PD and MSA. But one of the things is, do you expect to see different strains in different areas of the world, such as MSA in Japan, here? I've seen MSA in Kazakhstan and in Africa. Are they going to be different strains? Because the phenotype is subtly different.
That's a great question. I have to say that we have not looked into that yet. There's so many things to do that you have to go one by one. But I mean, I will expect that there are some differences. I'm dying to see MSA-PD, MSA-P, MSA-C differences. We haven't really looked into that yet. So we are, but again, we depend on having samples from different places.
So you'd be happy to receive?
Yeah, I'll be happy to get samples of those.
Skin, I know you're working on a plasma or serum assay too.
Yes.
Because we can't get CSF or brain from many of these.
Right. That's the problem. That so far has been limited because of CSF. CSF works extremely well, but it's more difficult to get. That's why one of the reasons we are moving with a lot of effort into something more easily collect. The other thing that we are doing that I think is also interesting is that we have some evidences that in different patients, particularly in PD, you have different strain-like properties. Not as different as PD from MSA, but you can actually differentiate them. So we're trying to see if people with different clinical manifestations of PD, they have different type of what we call sub-strains there. And the other thing that we're doing is to look at different brain areas from the same patients, because we think that maybe there are actually different strains depending on where in the brain. So it's a lot of things that we need to do, but we haven't done it.
I'd like to ask actually about what about LRRK2, where of course about 30 or 40% of them have synuclein deposits, 30 or 40% have tau, and 20% have neither. What do you find in those?
Yeah, in the Lancet Neurology paper, we communicate the result with LRRK2, and it was not surprising that we have a lower level of sensitivity in the LRRK2, it's like around 60% or so. I was expecting even less, but to tell you the truth, based on the histology. But that's what we see. A large number of samples, so it's there. What I think is the future is to couple several assays simultaneously. We have now a tau assay that is working pretty good. It's coming, and then have a beta, tau, synuclein, TDP-43 running the same samples to have a more clear-cut definition of. Well, from the point of view of Parkinsonism, it's very interesting that you can get the similar clinical symptoms with or without synuclein. I think synuclein is the triggering, but you can trigger the same type of pathology and in the brain by several other means. So.
Question here, and then Kailash.
Hi, I'm Kirsten Scott, a neurologist from Cambridge. The assay that you're describing is really good for prodromal diagnosis, but also have you thought about or tested samples that have been treated with alpha-synuclein targeting therapies. So, do you think the assay would be a good readout in trials where alpha-synuclein is the target?
Yeah, I mean, that's mostly being done in the company. And of course, the companies have they want to have everything very secret.
Yes.
The company who provide the samples. I don't get involved into that too much because it's so much work. I mean, basically, every single company that is developing trial is working with us, but all this work is being done in the company, not in my lab.
Okay.
So I'm not aware of all the results, but hopefully some of this will be published, and we will see what happens, because this is very important. What we want to do is to do a research study in the animal model to see if we take the animals and when we start to detect in the animal models, when before the pathology appear, and also what happens when we do treatments. So, yeah.
Thanks. Great talks, both of them. The MSA, the spreading is so rapid compared to the PD model which you showed. Why is there a predilection for certain areas? Why doesn't MSA spread, for example, to the cognitive areas and so on? Why is it restricted to those particular areas is one sort of question. And is your assay the similar or different from Nobuko Hattori's work, which was published in Nature Medicine? Because they say they can show it in the blood quite easily, and they show differences in the fibrils with MSA versus PD. So I didn't think you mentioned that, but maybe slightly different. So those are two questions to you. And to John, do you think MSA is genetic at all? So are we just harping down there? Are we giving lots of money to Henry for no reason?
I think there has to be, there has to be there will be genetics of speed of spread, for example. So there will be genetics, I'm sure. I think the largest GWAS is about 2,000, something like that. The Alzheimer GWAS, until they got to 2000, is when they started first to see signs, genes other than ApoE. So it seems like it's nothing, but actually Alzheimer's disease, apart from ApoE, was at that stage with these sample numbers. So it will come. It will come. Keep feeding Henry.
For my questions, the why different strains or different proteins will target different areas of the brain. This is the biggest, I think, question in the field for me, that we still don't know why attack different areas of the brain. We are trying to couple our studies of seed amplification assay with transcriptomics and proteomic analysis. For example, one of the things we can do now is we can extract the single cells that contain the aggregates, and then this is enough to detect with the seed amplification assay. So we want to analyze cells that have aggregates versus they don't have, or also from different distances using special transcriptomics. We hope, plus the genetics, of course, that we hope that will tell us what other proteins or genes are involved in the neuronal susceptibility in different areas of the brain. That's I think a biggest open question in the field. I think it's very important. The second point with the Hattori paper, have to say that we tried to reproduce it. We could not the results. I have to say that we have, like 3 or 4 years ago, we have this type of results, but we didn't publish it because it was not reproducible. So the thing is that especially for blood, the quantity is so low that you have to have a very precise way to treat the samples, because the plasma or serum samples are very complex, much more than the CSF, and you have to do something before you do the amplification assay, and that has to be very precise to be able to get the same result across different cohorts. So we got the result with some samples, very good, but when we tried to repeat with a different set of samples, we did not get the result. So we decided not to publish until we have something that is highly reproducible. So what I think it happened is that they got with one set of samples that they got good result, but then it remained to be seen whether they will be able to make it working with a larger number of samples.
Okay, we have time just for one last question. Take the microphone up to you.
It's a long trip. Thanks. Roy Freeman, I'm a visitor from Harvard Medical School, Boston. Those were two great talks for Prof Soto. So our lab is focused on skin immunohistochemical detection of alpha-synuclein. And one of the many advantages of skin immunohistochemical detection, and apart from the very high sensitivity within... Please. I'm using the mic. Our lab is focused on skin immunohistochemical detection of alpha-synuclein. And one of the many advantages of skin immunohistochemical detection, apart from the high sensitivity in MSA in the high 90s, is that it provides a quantitative measure that correlates with disease severity and therefore a potential pharmacodynamic biomarker measure of target engagement in disease-modifying trials, which I think we're all hopeful will be the way of the future. I'm wondering where seed amplification lies with respect to quantitation.
Yeah, that's another great question. We actually been working together with a group in Italy, the Donadio's group that developed initially, and working together to see if we can actually detect in the skin and correlate what they have by immunohistochemistry, what we have. And in terms of a quantitative, I didn't have time to tell you now, but there are ways to make the assay quantitative because the seeding process depends on the amount of seeds, aggregates you have. So you could, for example, if you take a seed that you produce in vitro in the lab, the same seeds that we amplified in the assay, and you put them in CSF at different quantities, you will see different kinetic of aggregation. So, the kinetic of aggregation correlates precisely with the amount of seed we start the reaction with. So it is quantitative. The big question is, and people assume in the field that the alpha-synuclein concentration of aggregates in CSF it's related to the disease severity, but it might not be. It might be that before the clinical phase of the disease, it reached already a plateau and doesn't change anymore. It's the biology of the disease, nothing to do with the assay. The high turnover of the CSF may also prevent the possibility to detect different quantities. So I think the assay is quantitative. The question is the disease also in a way quantitative? That I'm not sure.
Okay, with that, I'm going to close this session and thank the speakers once more. Hand over to Professor Houlden.
So I hope we can live up to that first session. I think we can. And continuing on the alpha-synuclein theme, which is clearly one of the important things in MSA, we have our first speaker, which is Steve Gentleman, who's a professor at Imperial. I've known him for a number of years. We've worked together on a number of chapters, which I'm very grateful for. And he's going to come and talk to us about MSA strains. So thank you very much, Steve.
Thank you very much. Thanks you to the organisers for inviting us along, particularly the MSA Trust, who are funding this PhD project that I'm just going to give you some of our initial results from. Tough acts to follow, as you say, but I think actually we're starting already to address some of those questions raised by both John and Professor Soto. So just a little bit of background. I'm the director of the Parkinson's Brain Bank over at Imperial, funded by Parkinson's UK. And we do get a fair number of MSA cases coming into the Brain Bank. I think we're just over 60 now. So we do have quite a lot to look at. Just your idiot's guide to the pathology of the alpha-synucleinopathies. We have Parkinson's disease dementia, and dementia with Lewy bodies, with the classic Lewy body pathology here in the nigral neurons. What's different in MSA is we still have some neuronal pathology, but what's definitive are these glial cytoplasmic inclusions in the oligodendrocytes. Now, what John was saying about going back to the pathology is very true, because we really are looking at massive clinico-pathological phenotypic heterogeneity. So not only in the clinical presentations, but no two cases are the same under the microscope either. There are variations. And really, this is what we want to concentrate on. So we've got the Parkinsonism. We've got the temporal evolution of the cognitive dysfunction in DLB and PDD. And then we've got the various, or the subcategories of MSA. I refer back to this Koga et al. Survey back in 2015. I'm not familiar with precisely what the details of this cohort were, but you had 134 diagnosed MSA cases, of which 83 turned out to be MSA at post-mortem. And as we heard earlier, DLB, PD, PSP, and others can be the diagnoses in life. MSA may be difficult, obviously, clinically at the early stages of the disease. So I hesitate to use the term prion-like, but I think we all know what we mean now in terms of spread and propagation of pathology rather than perhaps the transmission. And we go back to 2008, and in fact from studies from here, from the Institute of Neurology, among others, showing that the alpha-synuclein aggregates can form in neuronal grafts in Parkinson's patients. So as this at the time was put forward as a real evidence for a prion-type self-propagation and transmission of alpha-synuclein. And we have a fairly standardized spread of pathology through the brain in Parkinson's disease. It's not quite so clear in MSA. So there are all sorts of questions here about spread of disease pathology, differential vulnerability of different areas. Just very briefly again, historically, as we've already heard, Stan Prusiner now over a decade ago showed in the transgenic mice that you can transmit the MSA, whatever the agent is, to transgenic mice. And most interestingly there, perhaps, was he couldn't do the same with Parkinson's samples. I think slightly more contemporary is a study from Anke van der Perren and her group. And this, I won't go into great detail, but again, was using samples from the different alpha-synucleinopathies, samples from our labs, and they did a whole series of experiments, both in vitro and in vivo, to show differences between MSA and PD alpha-synuclein. That's the quote from the abstract there. I'm not going to go into details on this here. But this was really the background going into this project with Marcelina, who I should point out over there in the front row, who's doing all this work. Oh, there she is. Right. So our objective here for this PhD study was to look at the human postmortem tissue and CSF, but I should point out this is postmortem CSF, so it's not the same as what we were hearing earlier on. To look and see what we can see in way of differences from the other alpha-synucleinopathies. And we've broken this down into four main aims. So the first is back to the microscope, looking at different antibodies, different antigens on the alpha-synuclein. And I'll come back to why we went there first of all in just a minute. We've heard now a lot from Prof Soto about seeding kinetics of alpha-synuclein. It's something we're also starting to look at, and we've got some very preliminary results. But we also want to go back to the tissue in a bit more detail. It's not all about the aggregates at the end stage. We need to find out more about the process, we think, by looking at the monomers and the less aggregated forms of protein. So we're starting to use other techniques such as PLA, which is a proximity ligation assay, to try and look for areas of the brain where we may be seeing earlier changes. So just in the first one, antibodies, different antibodies, slightly different staining, different areas, different cases. Omar Al-Agnaf, who's renowned for his antibody production, certainly in frontotemporal dementias, etc., He has produced some alpha-synuclein antibodies, and Daniel Erskine up in Newcastle tried this on some MSA and PD cases and seemed to be seeing slight differences in what they were picking up in the different disorders. And this is the 8A1 antibody that we have here at the C-terminus of alpha-synuclein. And so we had a quick look at this to start us off. And yes, we also see a difference in the type of stain you get, but with different antibodies. So over here, this is a section of ponds from somebody who had an MSA. Clone 42 is our tissue bank, normal diagnostic antibody. It's one used generally around the world for the diagnosis, and as you can see, it's fine. We see the pathology. But the 8A1 was showing us somewhat more in some areas. Now, when they were first looking at this, it was felt that actually the 8A1 was MSA specific. I don't think it is. We do see overlaps with the other Parkinsonian pathology, but certainly there's something different about what's being stained with this antibody, and we've been trying to characterize essentially why are we getting a much more fibrillar presentation of the pathology in these cases. So we've had a look. Obviously, well, the most obvious thing is, well, this is just in the neurons. It's in the axons, the neurites. Well, you know, not that simple. Doesn't seem to be particularly prevalent in those neuronal components that you thought. Obviously, we're going to think it's going to be in the oligos, but the probe we've got on that is not quite ready, so I can't show you any oligos. But what I can show you is that there doesn't seem to be much of any colocalization here with microglial cells. This is the EBL1 pan microglial antibody, or with the astrocytes and the GFAP. So at the moment, we have got a little bit here with a neurofilament heavy chain. What we assume is a large swollen axon with the alpha-synuclein in here. So that's where we're at the moment. We're getting a whole series of other antibodies from OMAR to try and come up with a survey of the MSA pathology in different areas of the brain using these different antibodies. But a little bit of a side issue, what we noticed when we were going through our large cohort of cases now is the emergence of these rather novel lesions. I say they're rather novel. They have been reported in a couple of case studies now, particularly Gabor Kovac has reported these, and a Japanese group fairly recently. But we were surprised to see these, and I don't think Marceline will mind me saying, when I first saw this, I said, you've mixed it up with a tau antibody. Do you want to go and have another look? But she did, and she was right. This was the alpha-synuclein. So we're getting these ring-like lesions. They're really quite distinctive, and I was interested to see in the previous talk some ring lesions appearing in the animals, I think it was. And Daniela Petrova-Quayle is one of our psychiatrists who is going through the clinical records for the cases that come into the tissue bank. So for the tissue bank, we're generally a retrospective case review. And that's not easy for any of you who've tried to do it for the tissue banks here, because you don't know you're getting all the information. And frankly, cognitive testing, I don't think is routine with most patients with MSA. So there's not much hard and fast data. But she has gone through all of these clinical histories, and she's identified some with mild to moderate cognitive decline. And we're starting to see a match between those cases and the appearance of these hippocampal ring-type lesions. So more work here, but I think, again, there aren't that many MSA cases coming to autopsy, and we have a textbook idea about what the pathology is. But I think there's a huge amount of heterogeneity, not only in the clinical, but the pathological basis of MSA. More news on that. We've got a manuscript in preparation on those. The other thing there was, again, those ring lesions, P62. So this is a ubiquitin type inclusion. This is our pan inclusion antibody. No obvious link with TDP43, which is the other thing you would go to straight away in a routine diagnostic environment. So, I don't think there's much to say here. You've heard a lot in the previous talk about alpha-synuclein seeding kinetics. We're going through very much the same development, much earlier stages. Purification of the protein, and we've been doing this with Yu Yi, who is a protein biochemist in the UK DRI Centre over at Imperial. So doing the protein purification, and this is literally hot off the press yesterday. Some of our first RT quick results. And again, I think the first thing to say straight away is you can see heterogeneity. So here we have the purified protein fibrils, and here we have 4 different tissue homogenate cases from MSA patients. So we're just getting into this. As again you were hearing earlier on, there are always replication issues with this type of technology, so we need a bit more work on this. And ultimately, we've been doing this on tissue homogenates, but we also want to push this into the CSF that we have at autopsy as well. My worry again there is that I don't think anyone thinks a CSF assay can be single protein based. The co-pathology, the comorbidities, confounding pathologies in all these cases of a certain age, over the age of 60, there's always some Alzheimer change, not enough for cognitive decline. But I think one size does not fit all when it comes to looking either at the clinical presentation or the pathology of people living with MSA. The FRET cells, again, another aggregation assay, and we haven't got that working yet for the alpha-synuclein, but this is in the lab using tau, and we can see tau aggregates. So P62 again for the aggregates. Here's the tau DAPI, and here's the merged version of that. So you can use these FRET cells to have a look at the aggregation properties of the, what we will be, is the alpha-synuclein that we're actually getting from the tissue samples. I think, yeah, one more. Say this is early days. Marceline has not been doing this for too long, but is also already getting some very interesting results. Coming back to what are the early areas of pathology. Not easy again with MSA because it's not so predictable in its spread. If you go to Parkinson's, there's a Braak staging 1 to 6. If you have a patient who was Braak stage 4, you have an idea where it might be going, i.e. up into the cortex usually, and you can go and look at the cortex for early stages of disease progression, formation of pathology. Not quite so easy with MSA because it's not so predictable in its spread. On the other hand, we now have, thanks to Maria Otero Jimenez over there, also in the audience, she's been doing some PLA, particularly in tau, but she's also done some with the alpha-synuclein. So we now have the technique up and running in the lab to start looking, can we see earlier signs of change? And of course, we have people who've died at various stages of disease. Many, of course, are end stage, but we do have some who are perhaps slightly earlier in the disease pathology progression. So I'm conscious of time, and this is obviously very preliminary results because we've only really just started, but I think I'm glad to hear we're addressing the right questions according to Prof Soto and John. Thank you very much, and I'd just like to thank the Tissue Bank team, our funders, of course, both the all 3 charities for the Brain Bank and for the project. And this was the Christmas lunch last Christmas for the Tissue Bank. And I don't know why Javier wasn't there, but there he is anyway. Thank you.
Thank you very much, Steve. So thank you very much on that theme. Very nice talk. And we still stay on the synuclein theme. Focusing on one particular cell type, but we think could be important, but we'll hear all about it from Sonia Gandhi, who's a professor here. Thank you.
Okay, thank you. So I didn't introduce myself formally in the opening remarks. I work in a clinical service with Kailash Bhatia here at Queen Square and run a translational research centre. We call it the Movement Disorder Centre at UCL. And my research is at the Francis Crick Institute, just a few blocks down the road. So, you know, when we think, you've seen this a lot of times before, but of course MSA is this rapidly progressive and fatal sporadic synucleinopathy. And really what links it to some of the other synucleinopathies that I study is the idea that the alpha-synuclein aggregation occurs in multiple cell types, but particularly in oligodendrocytes. And we know that patients who have mutations in the alpha-synuclein gene, as John pointed out, the triplication, or shown here, a family with the G51D mutation that was seen by Henry and his team. The 19-year-old, the proband case, who has a very severe form of Parkinson's onset at the age of 19, but goes on to develop autonomic failure and dementia. And this is the post-mortem study of that individual who has widespread alpha-synuclein aggregation. And then in the fluorescence studies, you can see that the aggregates are also in the microglia, the IBA1 positive, the astrocytes, but in particularly also in the olig-2 or oligodendrocytes of the brain. So widespread aggregation, but particularly also in oligodendrocytes, is really what unites the patients with alpha-synuclein mutations and Parkinson's disease and MSA. But we became very interested in what oligodendrocytes are doing in Parkinson's disease as well, and that's because of a number of single cell studies that have emerged. So one thing we often don't think about, this is a UMAP plot where every spot is a transcriptome, and they're clustered by colour according to cell types. So it turns out that approximately 70% of the substantia nigra actually consists of oligodendrocytes. Now, oligodendrocytes typically myelinate neurons, but if we think about all the neurons that project from the dopaminergic neurons at least, from the midbrain through to the striatum, they're all unmyelinated and highly arborized cell types. So you've got a lot of oligodendrocytes in the midbrain, not myelinating, but providing other supportive features. And in these studies, what they showed, again, these are UMAP plots, and you're seeing clusters of cells. The oligodendrocytes in Parkinson's, at least, have a reduction in their number, while the microglia increase. So we know that there are glial changes happening in the midbrain in Parkinson's, and that as those glial changes occur, we're very interested in the mechanisms that are happening and underlying this. Furthermore, we've always thought about genetic risk for Parkinson's being in the dopaminergic neurons. That's the bar at the bottom on the left-hand side. But in fact, the genetic risk for Parkinson's is enriched in genes that are expressed in oligodendrocytes and oligodendrocyte precursors. You're seeing that in the red bar here. That's a study that looked at, for example, Alzheimer's risk. You can see very much in the microglia, but Parkinson's disease risk in oligodendrocyte precursors, oligodendrocytes, and dopaminergic neurons. And moreover, in those studies, changes in the oligodendrocytes are happening at Braak stage 1 to 2 prior to the changes that we see at Braak stage 3 to 4, positioning again that the oligodendrocytes may have a role much earlier in disease than we'd understood before. So what do I do in the lab? Well, what we do is we take patients with mutations that cause Parkinson's disease, and we program their cells, their skin cells, into induced pluripotent stem cells, and then we perform directed differentiation to try to generate the cells of the central nervous system in a dish so we can start to study their mechanisms. And the mechanisms that my lab is particularly interested in is why and how proteins misfold at the earlier stages, those oligamous seeds that become amplified in assays. How does the seed first form inside, and is that cell type specific? So we went on, this is a PhD student of mine, James Evans. So we've taken patients, we have 5 patients with the A53T mutation and healthy controls and some isogenic controls. And using a protocol that essentially overexpresses a transcription factor called SOX10, we can transform the stem cells into oligodendrocyte precursors, and then we can enrich those precursors using magnetic sorting so that now we have a culture that is very enriched for oligodendrocytes, and they're human oligodendrocytes, so we can begin to study those mechanisms. So I'm going to tell you about 2 particular mechanisms that we found that we think are of major interest in synucleinopathies. So for a long time, we've been studying how oligomers form inside neurons. And in this piece of work, we developed a single molecule FRET biosensor where you could take fluorescently labeled populations of the protein, add them to the cell, and track the structure and the size of the aggregates that form inside a human neuron. And you could even pass the lysates down a single molecule microscope so you could size each single aggregate one at a time, which is really sort of high resolution of what's happening inside a human neuron. And we reasoned we could do the same inside our human oligodendrocytes to see what's different. So these are little tiny aggregates. They're about 100 nanometers in size inside oligodendrocytes in a dish. You can see them whizzing around. They don't do this in neurons. You can see them moving between cells and passing up and down through the cell culture. What are they and why are they there? Well, when you use sort of more advanced microscopy, this is ISO microscopy, which is super resolution, what we can see is the alpha-synuclein in green is actually coating the surface of lipid droplets that are induced in the oligodendrocytes, which you're seeing in purple. And then we've gone on to do some correlative light and electron microscopy, which is where we pair the fluorescence with the electron microscopy, and we take Z-stacks through the whole cell. So you're seeing the purple lipid droplets, then you're seeing the FRET, which is the oligomers, and you can see them just forming as well on the lipid droplet membranes. So the ability to induce lipid droplets in oligodendrocytes by alpha-synuclein means that there's then a surface in which the aggregation can be rapidly accelerated, because we know that alpha-synuclein interacts with lipids, and it's that interaction that really drives aggregation. Well, we've looked at the oligomers that form in some detail, and this is from another paper that started to look at the structure of oligomers. But most importantly, what we find is that the sites at which they accumulate on these lipid droplet surfaces end up being disrupted. And that's because these oligomers, particularly they bind THT, they're beta sheet rich, and they're amyloidogenic. And in fact, they're able to interact with membranes and permeabilize them. And they can do something else. They can recruit what you're seeing in red next to the lipid droplet. You're seeing alpha-synuclein in green. Embedded between the lipid droplet is mitochondria around them as well. And peri-droplet mitochondria are also very unique. And then going back to looking at the aggregate in cyan there, you can see that the aggregate also is forming at the mitochondrial membranes, which we'd shown before in neurons, and again disrupting those membranes. So aggregate formation in oligodendrocytes is different to neurons and is probably highly cell-specific. And there are many, many features of oligodendrocytes that can accelerate the aggregation of alpha-synuclein. And quite importantly, it's not just lipid droplets that interact with mitochondria, as I've shown here and here in the electron microscopy, but also with lysosomes that also get recruited in this bundle of lipid droplets, mitochondria, and lysosomes. And we've been very interested in the idea that the peri-droplet mitochondria are actually abnormal. We've studied their function. And peri-droplet mitochondria are very important because they provide fatty acids, the lipid droplets do, to the mitochondria. And in this work here that we did, in collaboration with Stephen Gentleman as well, was to take early Parkinson's brains and look at the proteomics across the entire gradient through the brain. And what we found was actually that fatty acid oxidation was a major source of metabolism that happens in early Parkinson's disease. And in fact, one of the proteins that significantly regulates fatty acid oxidation increases in early Parkinson's disease and is located in the oligodendrocytes. So we think that oligodendrocytes might play a role in a metabolic switch that happens and therefore alters their support for neurons. But just going back to what we see in the human brain, we have a very large study. It's an ASAP-funded study of about $9.7 million now that involves a number of different groups. And what we're looking at is sporadic human brain. This is now from Braak stage 3-4. We've learned how to use some background reduction methods. You're seeing an astrocyte, in fact, being segmented on the left-hand side, and on the right-hand side, an oligodendrocyte. And inside the oligodendrocyte, those orange spots that are within oligodendrocytes are very, very early oligomers forming. So we actually know that these small aggregates that we see are also in human brain tissue. And we've done quite a lot of single-cell sequencing to look at oligodendrocyte populations. We see the populations of cells subdivide between neurons, glia, and then we can look at their substates as well. And quite importantly, the details aren't so important. What we find is in the oligodendrocytes, in the cortex of early stage brain, we're starting to see changes in inflammatory pathways. happening specifically, again, in oligodendrocytes. So they do play a role in Parkinson's. And what is this inflammatory phenotype doing, and what is the mechanism underlying it in the dish? So back to the dish then. When we do bulk RNA sequencing in our oligodendrocytes in the dish, we also see that the alpha-synuclein mutation activates inflammatory pathways, responses to virus, and defense responses to virus. And interestingly, something that we also see with all glia, that when glial states switch to become inflammatory in the human brain, they often downregulate their homeostatic processes. So these ones also downregulate myelination, and they downregulate their metabolic support. And we ask the question, well, what's underlying this process, and is this linked to alpha-synuclein aggregation? So alpha-synuclein in oligodendrocytes is interestingly largely localized to the nucleus, which has been reported previously. And what you're seeing here is the nucleus of an oligodendrocyte with alpha-synuclein staining, you can see with the DAPI. And importantly, the nuclei also reveal a large number of DNA damage markers in oligodendrocytes. There is a sensor of DNA damage called IFI16. It's a gamma interferon inducible protein. It's very important for sensing viral or foreign DNA. And it consists of a number of different domains that can bind double-stranded DNA. And most of the time, there are nucleosomes that prevent IFI16 activation so that we don't sense self-DNA. So we were really interested then to study this as a damage sensor in the inflammatory response. So here in the control cells, you're seeing IFI16 localized to the oligodendrocyte membranes. But this is in the A53 T cells, so the cells with a synucleinopathy that has a large number of aggregates in there. And there we see IFI16 translocates to the nucleus, where we think it's sensing that DNA damage that we're seeing in the oligodendrocytes with the synuclein mutations. And importantly, in the transcriptomic data, we know there are different isoforms of IFI16 that have PYD domains or DNA damage binding domains, and we see this massive shift into the transcripts of IFI16 that actually bind the DNA damage. So putting all of that together, I'm going to say what I think is happening in oligodendrocytes and synucleinopathies. I'm going to say I think they're a really fundamental cell type, and while we've taken a very neurocentric view, it's really important to understand the glial environment. Alpha-synuclein localizes to oligodendrocytes, both to the nucleus and the cytosol. As aggregation happens in the cytosolic environment of the oligodendrocyte, we see lipid droplet formation, and we see mitochondrial dysfunction and lysosomal responses, and that may alter the way in which oligodendrocytes are processing alpha-synuclein and releasing it back into the extracellular space. But aggregation within the nucleus may also induce DNA damage, and that may be sensed by the DNA sensor IFI16, and that will switch oligodendrocytes away from its supportive function of metabolism and myelination, and much more towards a pro-inflammatory and probably neurotoxic state. And if we can understand these mechanisms, of course, using Parkinson's disease, are these mechanisms also relevant in MSA, where we see a lot of oligodendroglial inclusions, and may they be mediating some of the progression that we see? And with that, I'm going to finish and thank my lab, who've done all the work, particularly James Evans, who made the oligodendrocytes, and all the funders and the patients and people who've donated cells and tissue. Thank you.
So to try and follow that up, we have Maria from Athens. It's very nice to have a Greek researcher come and present. Me and John have had a number of excellent Greek researchers work with us, both fellows and very senior people like yourself. And Maria's going to tell us about autophagy in MSA. Thank you very much.
So hello, I'm really honored to be here and stand amongst you to provide you an update regarding our MSA trust-funded grant related to the role of autophagy in multiple system atrophy pathogenesis. The previous talks provided a good introduction of what will follow. We know that MSA is a unique oligodendrogliopathy. As already mentioned, glial cytoplasmic inclusions generated mostly within oligodendrocytes are the key hallmark of the disease. The main components are, as we know, alpha-synuclein, but we also study a lot in the lab the role of tubulin-promoting polymerization protein, or P25-alpha, which under physiological conditions can facilitate the interaction between MBP and tubulin, which is required for myelination, whereas In MSA, we know that it redistributes from the from the myelin sheath to abnormally extended cell soma, and it can facilitate the aggregation of alpha-synuclein in vitro. And some recent findings suggest that it stimulates the formation of a more aggressive alpha-synuclein strain with a more enhanced neuroprotective, neurodegenerative potential. And in regards to alpha-synuclein, we seem to believe that mature oligodendrocytes do not normally express detectable levels of the protein, so the origin of the protein within oligodendrocytes is still enigmatic. Two scenarios, either an ectopic expression or the uptake of neuronally derived protein. So having as facts that both alpha-synuclein and P25 alpha aggregation within oligodendrocytes underlies disease pathology, together with Panayota, we question what are the cellular events that govern the seeding of alpha-synuclein specifically within oligodendrocytes, and what is the role of P25 alpha in this process. As already mentioned and described in detail before, we utilize the PFF alpha-synuclein model that enables to monitor the templating of the endogenous protein and the cell-to-cell transmission, and we inoculated rat oligodendroglial cells with such inoculates, either human PFS or brain-derived material, and we observed that under baseline conditions, no detectable levels of the rodent endogenous alpha-synuclein is present, where upon PFF addition, we have a strong recruitment of the rodent alpha-synuclein upon PFF addition, which resides in the formation of abnormal alpha-synuclein aggregates. These findings were further cemented by immunoelectron microscopy with using nanogold particles with different diameters, where we were able to show that both rodent and human alpha-synuclein coexist within these filamentous structures in the cytoplasm of oligodendrocytes. In order to assess the role of P25 alpha in this process, we utilized three different cell lines, rat oligodendroglial cell lines, the control line that express low to not detectable levels of both P25 alpha and synuclein, the alpha-synuclein, human alpha-synuclein expressing line, and the human P25 alpha expressing line. And when these cells were exposed to PFS, we were able to detect the pathology-related phosphorylated alpha-synuclein only upon an excess burden of alpha-synuclein, and to a higher extent when P25 alpha was present, suggesting that this protein plays a crucial role for the establishment of alpha-synuclein pathology within oligodendrocytes. We next move on to study further this phenomenon utilizing mouse primary oligodendrocytes, either from mice knockout for synuclein, wild-type mice, or using the PLP mouse model, which has the overexpression of human protein under the PLP promoter selectively on oligodendrocytes. As you can see, the seeding, as well as the formation of pathological species such as oxidized nitrated, occur only when endogenous alpha-synuclein is present, and together with that, the redistribution of P25 alpha network is present only when alpha-synuclein is there. In the knockout cultures, we don't see any such effects. And the impairment of the myelin basic protein network is also detected in the presence of alpha-synuclein, and it is higher, it is enhanced when we have overexpression of the protein in oligodendrocytes, whereas the knockout cultures are protected. When we assessed similar events in vivo by using wild-type and human wild-type and knockout for alpha-synuclein mice and inoculated them with PFS, we were able to see impairments of the myelin network which were present only in the wild-type mice and not in the knockout mouse striatum. So such data led the foundation for the continuation of this work, which is currently funded by Multiple System Atrophy Trust, to assess what are the manner of clearance of both P25 alpha and alpha-synuclein selectively in oligodendrocytes. To address this, we targeted the autophagy lysosome pathway, and mostly we focused on the two of the three pathways, macroautophagy and chaperone-mediated autophagy. There were four aims that led out our proposal. Due to time restrictions, I will mention some data from most of them. So they expanded from animal model cell cultures, cell lines, double cultures of neurons to oligodendrocytes, and I will present you some unpublished data that we have from human postmortem brain analysis. In regards to the data that we have for the clearance mechanism, we saw that when we downregulated the LAMP2A receptor, which is the rate-limiting step of the CMA pathway, or when we downregulated the ATG5, which is involved in microautophagy, then we had a strong increase in the signal that detects the endogenous oligodendroglial alpha-synuclein. And in regards to P25 alpha, since there was not much information about how this protein is being turned over in oligodendrocytes, we saw that lysosomal inhibition in particular, and not so much macrodophagy, but CMA was involved in the clearance of this protein, and this made us to believe that perhaps it has a motif that is being recognized by the HSC70 chaperone complex to be delivered for a CMA-dependent degradation. And through molecular modeling, we were able to identify this new motif in the presence of P25 alpha sequence. And in order for us to be 100% sure that it is a CMA substrate, we utilized an in vitro system of incubation of recombinant P25 protein with rat brain lysosomes. So as you can see, in the absence of proteins inhibitors, the protein is being cleared via CMA-dependent degradation, where when PIs are included in the mixture, this is inhibited. And as a control, we also utilized 2 different concentrations of recombinant alpha-synuclein, which is a bona fide CMA substrate. And as you can see, this hampered the clearance of P25 alpha protein. So having established that the ALP is involved in the clearance of both GCI main hallmarks like P25 and alpha-synuclein, we then question whether if we enhance this pathway, we can have a beneficial effect in the clearance of alpha-synuclein species. As you can see here, this is an example. We incubated cells with AR7, which is an enhancer of CMA, or rapamycin, which enhances macroautophagy. And as you can see, both the seeded endogenous oligodendroglial alpha-synuclein, as well the oxidized nitrated protein, which is formed upon PFF addition, are effectively cleared in this system. And when we assessed in the urea-soluble fraction of these cells to detect the abnormal conformations of the seeded, both higher molecular as well as monomeric species, we saw that both of these aberrant conformations were effectively mitigated. And at the primary mouse oligodendroglial cultures, we wanted to further validate such data. And as you can see, both P25 alpha and oligodendroglial alpha-synuclein are increased upon lysosomal inhibition. And in a therapeutic, let's say, approach, when we either enhanced CMA or rapamycin, we were able to decrease both P25 alpha as well as endogenous oligodendroglial alpha-synuclein levels, which paves the way for thinking of therapeutic targeting of the ALP pathway to counteract alpha-synuclein pathology in the context of MSA. And the last part, I will show you some of our most recent data in regards to whether we have alterations in lysosomal indices in the human MSA brains. Our study design is shown here. So we have obtained brain samples from the Queen Square Brain Bank. These are the numbers, and we took 5 different regions: frontal, cerebellum, occipital, white matter, substantia nigra, and coated nucleus, and then to have brain regions with different GCI pathology. We fractionated these tissues with using detergents with increasing extraction strength. Keep in mind that the most insoluble ones, protein species, can be detected in the SDS and the urea-soluble fraction. And with Western blot, we analyzed different levels and conformation of alpha-synuclein, P25 alpha, as well as lysosomal markers such as P62 and LC3, which dictate the autophagic flux. The CMA markers LAMP2A is 70, GCase activity and levels, as well as levels of LIMP2, and the PD-link genes ATP13A2 and VPS35 and cathepsin D. In all of the subsequent slides, the data are shown like that. So on the left, there is a representation of the different subtypes of MSA, and on the right, we have grouped all the subtypes together. As you can see, in the triton-soluble fraction of the cerebellar tissue, we can detect decrease in P25 alpha levels as well as in monomeric, which may indicate that this may migrate and they may be accumulating in the highest detergent fractions, and this is also happening. We saw that in the STS-soluble fraction, both monomeric as well as higher molecular weight phosphorylated alpha-synuclein is detected, evident mostly in the MSA-C subject, as well as higher and low molecular weight total alpha-synuclein levels. And this is the same situation what we detected in the most insoluble urea soluble fraction, where again we have higher phosphorylated alpha-synuclein as well total alpha-synuclein levels. So we have a strong pathology. So the question is whether we can detect alterations in lysosomal markers. As you can see, we have a strong increase of both P62 and LC3 2:1 ratio, which are both markers of autophagic flux. This indicates that an ongoing impairment of autophagic flux is happening in this tissue, and we also assess what happens on CMA. We know that when we have a dysfunction of one pathway, the other sometimes is overactivated to counteract the impairment of the one pathway. Interestingly, we observed an increased levels of LAMP2A, the mature form, the glycosylated form, and increased levels of HSC70, so increased CMA activity in this tissue. And which is also interesting, I believe, is that we also detected an increased trend for the ATP13A2 and VPS35. VPS35 is also, I'm sure you know, a member of the retromer complex, and we know that it is involved in the impaired trafficking of LAMP2A, ATG9, and cathepsin D. So perhaps the data that we obtain for VPS35 and LAMP2A would be of interest and need further investigation. When we assessed levels and activity of GCase, we didn't see any significant alterations in the protein levels. However, we saw a trend for increased GCase enzymatic activity in this area that harbors a lot of alpha-synuclein pathology, as I already showed you. Now, if we try to compare the data we obtain from the cerebral white matter to the frontal white matter, you will see that in this tissue we have a milder pathology which is accompanied by a milder differences on lysosomal indices. So in the triton soluble fraction we didn't detect significant alterations. We have phosphorylated alpha-synuclein but detected only in in the STS soluble fraction, only 2 out of 20 cases. The same in the urea soluble fraction. In the case of cerebellum, we had most of the cases that displayed pathology. And interestingly, in regards to macroautophagic markers, we can see that P62 seems to increase together with the LC3 2:1 ratio, so a more global dysfunctional macroautophagy. CMA markers do not significantly change, so there's not any significance so far. And the trend, the picture for ATP13A2 and VPS35 is the same. And by measuring GCase activity in this tissue, in the frontal white matter, we detected a trend to decrease. And since we have the data for the GCase activity in all brain regions, I will show you this now. So we have this interesting, situation in the cerebellum. We have an increased MSA in MSA-C people, the GCase activity, and in the substantia nigra in MSAP. So this increased GCase activity in areas harboring the highest GCI pathology may reflect a mechanism to counteract regional alpha-synuclein accumulation. So if I may summarize the data that we have from the human study, there seemed to be a correlation between the detection of abnormal alpha-synuclein aggregations with alterations of lysosomal indices. Of course, we need to further elaborate on this data, and we need to include the remaining 3 regions, and we need to do correlation analysis between the alterations and lysosomal markers together with synuclein conformations and disease severity. And of course, we need to pursue further this project, and if we can obtain additional funding, we hope to analyze better alterations on the LP pathway in the periphery by utilizing PBMCs or exosomes from serum and plasma derived from MSA patients and controls. And also we want to study more the mechanisms by utilizing a novel 3D organoid system that we have established in the lab. We have some very preliminary data with a small cohort that we can see that we can detect with Western blot some alterations in these macroautophagy markers. Of course, these need more clarification. And this, I'm ending. I would like to thank the people that contributed to this work, especially Panayota and Fedra, all the people in the lab and collaborators, our fundings, and of course, Multiple System Atrophy Trust that enabled to pursue this endeavor. And thank you for your attention.
So thank you. Now we have our fourth rising star, Valeria, who is going to tell us about diagnostic and genetics. I've known Valeria for a number of years, and she's continued to do well and push the boundaries in MSA, as she'll tell you about here.
Thank you. And yes, this is the last session, the last talk of this session, but we're coming back to genetics. As a lot of the previous talks mentioned the importance of actually having the right diagnosis for any of the research that follows. I thought it would be interesting to bring in the topic and to launch a conversation on the role of genetics and genetic testing in the MSA clinic. And good. I'll just talk then. So first, how we diagnose MSA clinically is based on diagnostic criteria. And briefly, they rely on clinical signs. And depending on how many of those you have, you get a more certainty of the diagnosis. So at the beginning, if you have for example, a combination of ataxia, Parkinsonism, some autonomic features, that is kind of, it tells you, but it's not moving. It's moving. So there it is. So if you have a few of these symptoms, you'll have a possible prodromal MSA diagnosis. And then if you have some additional supporting features, if you have one of supporting features, you would get a probable diagnosis. And then if you have two, for example, you'll have a clinical established MSA diagnosis. And at this level, we kind of have some MRI biomarkers to have a more certain MSA diagnosis. You can only have a definite diagnosis at autopsy after the patient has died when you have this GCI inclusions. And this is how a MSA patient looks like, and you can hear the voice.
British Constitution.
British Constitution.
This high-pitched and dysarthria, this is a patient with mostly cerebellar syndrome. And my talk will actually focus a lot on the cerebellar phenotype, because there will be a lot more on the Parkinsonism later. And you'll see the clinical signs that help us diagnose cerebellar ataxia or cerebral syndrome with the eye involvement and the limb ataxia later on. I'll just, for the non-clinician in the audience, I'll just leave this for a few more seconds, because we have other variants that I'll talk about later, just to give you an opportunity to compare. So these are the cerebellar features, and you'll see in a minute some of the Parkinsonian features, which are less pronounced. So this patient has a kind of a mixed phenotype, but predominantly cerebellar.
I've had trouble focusing at distance lately.
I leave the voice because it is quite typical of MSA, and this is the Parkinsonism. And you'll see that is mostly on one side. Yeah, and the patient is in wheelchair. It just shows the severity of this condition. But they are. Next slide. They are other kind of variants of MSA that have been described with some cases that have Parkinsonism for a very long time before they develop autonomic features. And there are cases where they have this isolated Parkinsonism or isolated cerebellar syndrome before the autonomic features appear. And in average, it takes about 2 years for a concomitant motor features and autonomic features to coincide. And this gives rise to kind of this variant of MSA. But this gives me the opportunity to kind of open this somehow controversial topic of genetic testing in the MSA clinic. And I think this will help us clarify some of these variants that we have in the literature. So together, Professor Henry Houlden and Dr. Go, we do the MSA specialist clinic at Queen Square. And when a patient is referred to us, we frequently include some genetic testing in our workup, in addition to the usual diagnostic workup for an MSA patient. And I will explain why we do that. Well, we start with spinocerebellar ataxia, and this and many other groups have shown that there are a number of patients with spinocerebellar ataxia that would fit the MSA diagnostic criteria. And why is this number so high? Well, SCARS, this is a big group of genetic conditions that have as primarily as their presentation the cerebellar ataxia. And when a patient with cerebellar ataxia has, and the SCAR gene mutation has associated autonomic features, that could really easily be missed as a MSA-C. And some of these SCAR genes, they also can have neuroimaging signs that are frequently seen in MSA, such as hot cross band, for example, in SCAR2, 3, 8. And then there are other scars that can present with Parkinsonism without cerebellar syndrome. So again, those ones could easily be misdiagnosed as MSA-P, for example. Now, as I said, this talk will focus mostly on the cerebellar phenotype, and our group has shown a few years ago that mutations in expansions in RFC1 gene cause condition called CANVAS, which presents with cerebellar ataxia, neuropathy, and vestibular failure. And this is a patient with a CANVAS RFC1 expansion. And you see the cerebellar phenotype, you see the increased reflexes, pyramidal syndrome, and then at the end, just to warn you to look at the eyes, you will see the vestibular reflex, which is abnormal. Okay, this is just the cerebellar signs, and that's it. This is the abnormal VOR. And so this disease is very slow progressive, has cerebellar ataxia. Patients become wheelchair bound very late in the disease, and compare that with the MSA patient that I showed you already in a wheelchair within a few years from presentation. But all these patients, that's important that all these patients 100% have neuropathy associated. And other important features are the vestibular impairment, which is in almost all of them, and many of them have the spinal cord atrophy on MRI. Now, because of this combination of symptoms of cerebellar ataxia and possible autonomic features, we screened all of our clinical MSA patients for RFC1 mutation. So we did the usual protocol with flanking PCR, repeat prime PCR, and southern blot. And we found 3 patients from our clinic who had a biallelic expansion who was in keeping with an RFC1 positive diagnosis. And then we screened all the path-confirmed MSA that we had available, and over 200 patients, 300 patients, and none of them had the expansion. And so this is dual pathology, just the mimic, a phenotype mimic, but I'll show you how easy it was to think that this could have been MSA. And this is the video of the patient. All 3 patients presented in their late 60s. They all had a combination of cerebellar and Parkinsonism with autonomic failure. The autonomic failure was mild to moderate, but in early diseases that could be the case in MSA. And they had abnormal DAT scans and a negative family history. So at the time, they did fulfill the diagnostic criteria for MSA. What was, however, different between a typical MSA is that this patient, for example, the one in the video, they had they had a peripheral neuropathy. However, in this age group and neuropathy being so frequent in general population, this can easily be just thought as 2 diseases. And one of the patients had abnormal VOR, but unless you look for the abnormal VOR, you will not know it's there. So it's just to remind you that to not forget to check for it in a patient with ataxia. But next, I want to show you this patient who presented with Parkinsonism and autonomic failure, and the autonomic failure was very severe, and it fulfilled the diagnosis, it was consistent with an MSA diagnosis of autonomic failure at the time. But this patient, again, had peripheral neuropathy. So just want to, I hope I convinced you that it's worth checking for these expansions, both in MSAP and MSAC, because they can be difficult to diagnose, especially in the early disease stages. But next, I'm going to this new gene, the FGF14, which is predominantly expressed in the nervous system, central nervous system, in the cerebellum. And it has been shown that the pathogenic range, above 300 repeats, is fully penetrant. Under 250 is intermediate penetration, is normal. And then you have an intermediate range, which we still try to define what it means for disease. Now, there are some control cases or cases without clinical signs that have pathogenic range expansion. This, I think, are the pre-symptomatic subjects of this late onset disease. The phenotype is a very, very slow. It's even slower than the RFC1. Patients start with some episodic symptoms, then become more permanent. It's predominantly defined by ataxia, cerebellar ataxia, but they have some additional symptoms such as neurogenic bladder in about half of them. Walking aids and wheelchair only happens after 15, 20 years, so very, very slow progression. And other associated signs, neuropathy, spasticity, but urogenic bladder really is one of those that could make people think that a combination of ataxia with bladder involvement in this age group might trigger this possible MSA diagnosis. So because of that, we looked again at our cohort, but it is time we screened 600 and almost 700 patients, of which 3/4 were path-confirmed MSA, but only 200 of them were from clinic, and these were all patients who had onset after 30 years, absent family history, and who all have negative for the SCA genes, and we included both phenotypes, MSAC and P. And we did a protocol that was published for the validation of this expansion, and we found expansions in 18 cases. This is well above the ones that you'd expect, the one that's published in the healthy controls, and actually we have data now from yesterday until today. We have data on 1,000 healthy control, which shows a frequency of 0.1. So we have more than in controls. This is in MSA. But what I want to show you here even more is that 10 of the 18 are path-confirmed MSA. So these are pathology-confirmed MSA with standard diagnosis of GCI who also have expansion in FGF14. Now, phenotype. The phenotype is really difficult to, like this, these are all MSA patients. This is not a misdiagnosis. These are all MSA patients. They have all the features, Parkinsonism, cerebellar ataxia, autonomic dysfunction. They have REM sleep behavior disorders. They have stridor. They have pyramidal signs. And as I say, these are majority of them are path-confirmed cases. And the clinical cases, they all still fulfill the current diagnostic criteria. They were, in terms of phenotype, they had both MSA-P and MSA-C phenotype, and most of them were mixed. The sensation, cognition, behavior was normal in all the expanded cases. But what was interesting was mostly the survival of these patients. The patients without MSA patients without FGF14 and MSA patients with FGF14, there was about 2 years of difference in their survival, which for MSA, this means a lot. Now, this is how these patients present as a patient with path-confirmed MSA and FGF14 expansion. And this is the two videos are 3 years apart. So this you'd see, like in the first video is the phenotype was predominantly MSA-P, so the Parkinsonism was predominant. You'll see in the second video is actually there is quite important cerebellar features associated with that. And these are path-confirmed MSA.
Close your eyes really tight.
It's good enough. Parkinsonism is clear. Then in the second video, there's a lot of eye involvement, the eyelid apraxia. The patient is not falling asleep. This is the eyelid apraxia. The voice, you can't hear it because the voice is affected. It's like severe dysarthria and dysphonia.
British Constitution.
With all the supporting features of pyramidal signs, Babinski sign, polymyoclonus, and the cerebellar here. It's quite clear compared to the baseline. So this rapid progression and new features as the disease progresses. Now, this is just a very brief picture of the late onset ataxia presentation in the MSA clinic and what might help differentiate between them. But it can be very hard unless you test for them, especially in the early stages. We are trying to kind of get this table more clear and with more information. And this leads me to the last point I wanted to say about what we do in our MSA clinic. We invite all the patients to join our research projects. And two of them that have genetic components, I'll just briefly mention here. This leads me to some of the points that John Hardy was mentioned today. We have collected now 1,000 pathology-confirmed MSA cases for our GWAS that we want to do on survival and disease. So since they are pathology confirmed, we have information on their predominant pathology subtype, so SND or OPCA or mixed. Because they are pathology confirmed, we are trying to get as much information on survival because we know the age of death. So getting back to getting the age of onset on 1,000 cases, that was a challenge. We have finished the data collection, the genotyping, the quality control, and the imputation. And we are actually right here at this stage. So this is a really cliffhanger before the tea break. And all these cases have been screened for rare variants and known expansions, and that's the data that I've presented previously. So I wanted to just point out that even in a pure path-confirmed cohort, it's still important to look for the dual pathology, because this is not unheard of, and we're seeing more and more of this dual pathology and other genetics that could influence AGUS, for example. And last slide is our ongoing natural history study that has also genetics, but a biomarker, strong biomarker component, and we collect samples from, this is a multi-center UK-wide project that has a longitudinal and a cross-sectional arm, and there will be a new arm, the Express study, that's coming soon for even earlier stages of the typical Parkinsonian syndromes. And we collect samples for biomarkers, for genetics, and for natural history. And we are looking forward to work together with you. And if you have patients to refer for either genetics or the prospect study, we very well encourage you to get in touch. And yeah, I would like to thank everybody for, from our team, from the Prospect team, and our international collaborations and brain banks around the world. But mostly, I wanted to mention that if you, after this talk, if you can think of any patients that maybe have some features that are not typical for MSA, in addition to their MSA diagnosis, or have an unusual disease progression, either very fast or very slow, to get in touch with us, because we continue to screen for these genes and other genes that I have not mentioned yet, and that might bring some clarity to these variants or outliers that are in the MSA clinics. Thank you very much.
Good. So we have a few minutes of questions. How many minutes do we have? 4 minutes. One question each. One question each. We can... But there's also the tea break for questions too. So, Claudio and one at the back. So Claudio, you first, and then Will's at the back.
Yeah, very nice talk, all of you. There is not much time, but Sonia, I wanted to ask you, these videos that you show of the movement of the aggregates are beautiful. How do you look that? What do you stay for? How do you make it?
Yeah.
Sorry.
With that, what we did was add fluorescently labelled using an A90C mutant of alpha-synuclein. We know the labelling at the 90 residue. And we have it Alexaflor labelled, so not a fusion protein, but we're adding populations of 488 and 594, and then we're following the FRET signal, so the energy transfer. So we know that anything we're following is an assembly. We actually sort of know the size of the assembly as well. And so we're imaging there recombinantly added. Obviously the endogenous, much harder to study, and then just live. And it's quite interesting because if those precede the formation of the larger inclusions, well, at some point they must become static and localized in the cell environment. But we don't see that sort of behavior in astrocytes or neurons, interestingly. So I think there is something quite unique about the oligodendrocytes. But a lot of those are moving on lipid droplets, I should also say. So there are mobile components of oligodendrocytes, and lipid droplets are one of them. So, yeah.
Thank you. Will, do you want to shout?
Yeah, sorry, I hope
you've got a loud voice.
Viorico, great talk. Thank you very much. I'm just interested in your thoughts on the SCAR 27B intronic, because that's what you were referring to. Do you think this is causing a sort of more widespread splicing issue, or do you think there's potentially DNA mismatch repair issues that could explain the enrichment?
we don't know yet. This is literally working on this right now. What I wanted to show for now is that the dual pathology exists. What it means for the MSA disease, we don't know. And if it is related or not, like a susceptibility for having this genetic diagnosis, does it make it more susceptible to MSA? I don't know. I don't think so. But they're not rare, and this one of the genetic conditions that is so frequent in the MSA pathology cases. We've never seen this before, like the association of two diseases with MSA, another genetic disease. So I don't know. We need to do more functional work on this. For now, I just wanted to make people aware that it is frequent.
So I think it's partial loss of function, as I tried to get Viorica to say just then, but she didn't say. She's not committing. But the cases that she showed, they're the MSA cases that often have very brisk jerks and upgoing toes. And this is a variant, quite an aggressive variant in some of the MSA patients that we see. But those ones, and some of these patients, I even imaged their neck on this lady more than once, because I thought there must be something else there. What am I missing? And she has this. I remember the lady distinctly. So it is an interesting group. And if there's no other questions, I have one for the more kind of pathological kind of experts here, particularly Steve and what Maria thinks are these ring lesions. They're very interesting. I mean, I've not seen that before. This is brand new. And you think it occurs with the more cognitive patients. The rapid, or those patients that we have around in clinic for 16, 17 years, or don't we quite know?
I'm afraid we don't have anywhere near that subtlety on this yet. We're still not entirely sure what they are. They're not exactly perinuclear, so it's unusual. It's not the same as other like FTLD inclusions. They just have a quite distinct morphology. So need more work, and that cognitive association is very loose at the moment, and we need some more work on that though.
One quick question for Maria. Did you measure glucosidase? You got GCase.
Yes.
What did you get?
It was on the data. So we measured in the different regions, and what we see, I mean, we were hoping to have a more, clear, let's say, connection between alpha-synuclein accumulation and GCase, let's say, levels and activity. So we were expecting to see the opposite. Since we have brain regions that have a lot of synuclein pathology, we would expect to see decreased GCase. However, our data so far indicate the opposite. So in the MSA-C cerebellum, we detect more GCase activity as compared to the other subtypes. And in MSA-P, we also decrease more GCase activity, whereas the levels of the proteins, either LIM2 or GCase, do not change. So this is an interesting finding, I think. Wow. And we would really like to see what happens. If you can measure this, because we measure this in PBMCs also. So we are using these assays to try to see what happens in the periphery. So it would be nice to see if this changes over time, because in the human postmortem samples, as you know, it's the end point, so you're not sure what was actually happening before and what is happening now. And this is why also the lysosomal fluctuations are of interest, that different responses we get for macroautophagy and CMA pathways, and they also differ between the different GCI-affected regions. And this is also that we need to investigate more and correlate it to synuclein pathology.
Okay, I'd just like to welcome you back to the next session. So my name is Chris Kobolewski. I'm a consultant neurologist in Manchester and trustee and chair of the scientific advisory panel of the MSA Trust. And I'm very thankful to the MSA Trust for asking me to chair this session, which is on MSA research from patient to bench and back to the patient. It's really important that we translate our research in that way, and I'm looking forward to hearing what the presenters have to say. So our first talk for this session is Dr. Amanda Hesselgrave, who is from the Biomarker unit in UCL. She's worked for many years on biomarkers in this unit and was involved in setting up the original biobank at UCL. And Dr. Hesselgrave is going to talk to us about biomarkers and their relevance to MSA. Thank you.
So hello, and thank you very much for inviting me, and thank you for the introduction, Chris. I'm actually going to talk about I'm actually going to talk about what you need to think about when you want to collect samples for research. And that could be because you want to set up a biobank. I'm just going to say, after this talk, don't ask me to biobank your samples because we actually don't do that anymore. That's not in the remit. But anyway, so I work in the Fluid Biomarker Laboratory, which is part of the UK DRI at UCL. We're based at Queen Square House, just across the way. And what we do is predominantly measure biomarkers in fluids. We provide the expertise for people who are looking to set up biomarker projects. We develop new assays where there's a need, or we try to, all to understand dementia better. So the Biomarker Factory is part of the Fluid Biomarker Laboratory. This is funded by the UK DRI as a resource, really, to the community, to give people the expertise and the equipment required to measure the biomarkers that were really going to help us with diagnosis and treatment. And so I co-lead this with Professor Henrik Zetterberg, who is probably the most famous biomarker scientist in the world, I think. And we have Rhiannon, Elena and Owen working with us to make sure that all these projects get done. So what samples should you be collecting? Obviously, I'm going to talk about fluids and really going to concentrate on CSF and blood. CSF is the obvious choice. When you're looking at brain diseases, you are more likely to find information from CSF than any other fluid. And what our advice is when you collect it is collect it into polypropylene tubes. All of the things that we look at are, well, a lot of the things that we look at are aggregative, as we know. We've been talking about alpha-synuclein all morning. And then we've got SOPs we can send to people. If they want to start a project, we can send you an SOP on what we would do in that situation. But basically, I've laid it out here. Centrifuge and aliquot. Think about aliquot sizes, because how many things do you need to measure? How many of those are going to be measured at the same time? Do you want freeze thaws? Does it matter? I mean, and actually people often come to us for advice on that, like, you know, we want to measure this. Does the freeze thaw amount matter? And we can give advice on that if it's been looked at. It's obvious, it seems to me. So yeah, aliquot doesn't want to be too small, doesn't want to be too big. But CSF, although everyone talks about the specialist doctors or practitioner that needs to actually do the lumbar puncture, and the fact that it has to be done in a hospital, it's still the gold standard for amyloid measurements, for example. And as we were told just earlier, although the RT-QuIC or the seed amplification assays are being worked up in blood, as far as I know, as far as I've seen, I haven't seen that that's actually working well yet. So CSF is still something that a lot of people want to collect. Okay, so then what about blood? When it comes to blood, you need to think about whether you want serum or whether you want plasma. And obviously, the way that you process those is just slightly different, but mainly your choice of tube. When it comes to plasma tubes, there are a number of different types of those, and it does need some thinking about, or at least you need to think that you're not going to change halfway through a project, which is something that someone asked me about recently. If you want to collect DNA, you can take that from EDTA plasma. I'm not a DNA expert, so that's where I'll leave that. Likewise, RNA, but there are PAX-gene tubes for that as well. How do you choose what blood you're going to collect? Most of the projects that we do, most of the markers that we measure are actually kind of worked up in EDTA plasma, and normally it's K2. We can use serum, and like if people have got serum banked and that's what they've got for a project, well, that's fine. I think we find lower values in a number of analytes, but sorry, I've lost my train of thought there. We find lower values in a number of analytes, but for most, we can measure it. I think the crucial thing is that you don't measure serum and plasma in the same project for the same marker, because there will be differences there. So you just need to keep it consistent. You need to check if your additive, if your anticoagulant is going to affect your analysis, thinking of something that's calcium sensitive like S100B. And also, as I say, the levels can be different depending on your anticoagulant. Don't mix and match. Now, there are some really good papers actually that are out there, and most of them are about Alzheimer's disease, because that is obviously the biggest kind of neurodegenerative dementia disorder. But this particular paper, they've done lots of different kind of scenarios to check, to see if we can standardise collection of biomarkers. And they come out with a recommended workflow for blood sample handling, i.e. using K2, the time to centrifuge, the amount to fill your tubes with, and what long-term storage to have. They say -80 here. I'll mention that in a minute because that's not necessary all the time. And we need to consider optimal processing is what you're aiming at, but it doesn't always happen. But some proteins, it's really, really crucial. And this is going to be the aggregative proteins. These are the ones that give us a problem, are going to be more difficult to measure, and will stick to the tube so you lose it before you've even done an assay. Then moving on, and this is of interest to many people working in the field at the moment, is remote testing and screening. I mean, particularly where you've got therapy available or becoming available, you really want to screen your population. And people are working on blood and plasma cards to test and validate actually being able to measure useful biomarkers in these. Now, this paper just came out 2 weeks ago, I think. It's a bit disingenuous because it says dried blood spots in the title, but it's actually dried plasma spots that they've measured. Plasma tends to be easier to work with, plasma spots, than the blood because of all sorts of interfering proteins, cells, membranes, etc. So, but that is a big area of research right now. Also, Lauren Byrne, who works in the Huntington disease group, has been working with her team on capillary blood, actually comparing blood from finger pricks into the tiny little tubes to measure various neurodegenerative markers. And actually, this paper is on Rrisks at the moment. I think it's in review at JAMA Neurology. But they show that NFL, neurofilament light, one of the most widely used neurodegenerative markers, is stable after delayed processing for 3 and 7 days. And that it shows the same disease groups as venous, i.e. we can use it as instead of. So that's really quite encouraging. Then talk about storage, which I alluded to a little while ago. It used to be that ultra-low temperature freezers were set at -65 or 70, and only in the 80s, 90s did the producers of them start to advertise lower temperatures. Now, this costs an awful lot in energy use, but there isn't any evidence that it's improved sample stability. And so even time to freeze thaw, I think I say in this line here, I say, if you've got a freezer at -80, you get 35 additional minutes before reaching -20 in case of a complete power failure. So all of our freezers in the biomarker team are set at -70 now, and I don't see a real reason why we would change that now. Now the small things. Consistency is what we need to do. When you're thinking about a project for biomarkers, you need to have an SOP, which were designed for this, as I mentioned there. You need to follow the same SOP every time. That's going to give you the best results at the end of the day. You need to think about your tube labelling. I mean, for us in our lab, because we're research only, we absolutely don't want any patient details. We just want an identifier on that tube. And we need things with freezer-safe labels. I don't need to probably go into the number of times I've sat on the bench just looking at all the labels floating in the ice, which is not handy. Don't handwrite on tubes because we can't read your writing. And then database the samples as soon as possible so the information is captured, or the information that you're allowed to have is captured. And we've always really relied on Excel to do that. You can pay a lot of money for fancy databases, but Excel has always come through in the end for us. So now, really briefly, I'm going to talk someone requested this, and I think it was Viorica, that I talk about NuLISA, which is the newest technology available for multiplex biomarker measurements that we will be getting in our lab in March. So beginning of March, that comes to us. And basically what it is, it's a proximity extension assay that uses 2 antibodies to every analyte that you're searching for and pairs it to DNA to form an immunocomplex that's barcoded that can then be detected by next-gen sequencing. This is much more sensitive than our most sensitive assay platform that we have at the moment, the Quanterix Simoa. And the reason for that, or what would seem to make sense as the reason for that, is that they have a capture-release system that allows them to wash away almost every single part of the background. And if you've got a low background, then you get a higher signal. And this is actually proving to be, as I say, from samples that we've sent to the company, but better than the Simoa. And here they just show, this is some of their publicity material that they showed me, for example, with IL-4 or P24, showing the blue line, the actual, the better sensitivity you can get using this technology. And the thing that we're most interested in at the moment is what they call the CNS disease panel. And the reason we're interested in it is because it has all of the analytes that anyone comes to me asking for, all in the one panel. You can measure them all at one time. And it's the first one here, Alpha-Synuclein. Actually, this is quite an old slide, in the panel that's available now, there is Alpha-Synuclein and then a aggregated Alpha-Synuclein assay. I can't say at the moment how good they are. I haven't seen them in action, but obviously there's people in this room who are going to have samples that might be that will give us a lot of information about how well this panel actually works. As I say, we've got, oh no, what did I do? I don't know what I've done. Anyway, in that panel, we've got all of the phosphatals, which if you're in Alzheimer's research is just a dream come true to have that with neurofilament light, GFAP, and the amyloid peptides. So if anyone is interested in this technology, I am more than happy to talk about it or meet or whatever, and we can see if we can do some collaboration. So thanks very much for listening.
Okay. Many thanks, Amanda, for that talk. We'll come to some questions at the end of the session, but I'd just like to welcome the next speaker, which is Professor Nicola Pavese from Newcastle. So Nicola is the Professor of Clinical Neuroscience and Director of the Clinical Aging Research Unit at Newcastle. His research interests include DBS in Parkinson's and also works in atypical Parkinsonism, is a collaborator in the PROSPECT study and has extensive experience with PET imaging in particular. And so Professor Pavese is going to talk to us today about neuroimaging markers for MSA diagnosis and progression.
Ladies and gentlemen, good afternoon. I would like to thank the organizer for inviting me to give this talk on neuroimaging tools in MSA. I will be sharing the talk with my colleague, Dr. Jacopo Pasquini. I will start talking about the conventional neuroimaging finding in MSA, which you probably already know. And then Jacopo will present the data and the results of an investigation that we are doing in Newcastle. Short time, so I will mostly focus on structural MRI and on functional MRI, PET, and SPECT. Let's start with the MRI. In early stages of MSA, like in Parkinson's, the conventional MRI is usually normal. But as the disease progresses and you start having more established atrophy in the brain, you start seeing some characteristic features that can help to support a diagnosis of MSA. This is, for example, MSA-P, and the most characteristic findings are atrophy of the putamen, which reflects in hypointensity, so reduced signal in this area. But you can also find hyperintensity, particularly on the lateral aspect of the putamen. When it's present, this is called putaminal slit sign, and is quite characteristic of this form of MSA. In MSA-C, you have already heard about the hot cross band by Eureka. This is related to this hyperintensity that you see in the pons. Is a cross that recall the hot cross band. And this is related to the selective atrophy of the transverse pontocerebellar tracts and also the median pontine nuclear raphe. It's not specific. You have heard that you can also find that in patients who have spinocerebellar ataxia. You can also look at images in a sagittal way, and you can see here in MSA-P again that there is a flattening and atrophy of the pons. There is an atrophy of the cerebellar, and there is a dilation of the fourth ventricles. And that can be useful to differentiate MSA from PSP, because in PSP the atrophy is more in the midbrain. So as you can see, the pons is preserved, while it's the midbrain that is affected. Now there are a lot of softwares that enable us to measure with planimetry to have different index that can help in the differential diagnosis of Parkinson from MSA and PSP. Are these clinically useful? We said that at the beginning they are not present. This study has, you know that there are recent criteria proposed by the MDS Society to help with the diagnosis of MSA. Probably Michele will talk about this afternoon. This study looked at validation of this criteria. And what they did, they assessed the autopsy in over 250 brains with MSA and over 100 patients with non-MSA. And you can see that here are the hypothetical criteria. There is a clinical, A is core clinical criteria, B is 2 or more supportive features, C is the imaging, mostly the MRI, and then we have D, that is the absence of exclusion criteria. And they have checked different combination of this hypothetical criteria. And the interesting thing is where the C appear, so where the MRI findings are there, you notice that there is an increase of specificity, so suggesting that MRI can be very important to increase the specificity of this criteria of the diagnosis. What about progression? There are studies that have shown that there is a brain atrophy that can be followed up over time. This is studied by the quite recent by the Innsbruck group. They have looked at 43 patients with MSA and PD patients. They have looked at the whole brain and all different structures, and you can see that while in Parkinson's the annual rate is very low, in MSA is actually quite high, particularly in structures that are known to be affected in MSA. And interestingly, the pons seems to have a greater rate in MSA-C compared to MSA-P. So that could possibly be useful to follow progression, should be checked if can be useful in following up clinical trials in the future, as we really need bioimaging, wet biomarkers of disease progression. You have, Veronica also already mentioned the amazing Prospect UK study. This is a study that is following up progression in patients with atypical Parkinsonism, including MSA. In this specific study, the team has looked at progression of different factors, clinical, cognitive, and also imaging, to see if that can help with the sample size in clinical trial, so implication for the trial. And as you can see here, the most important part is that the MRI findings are able to reduce the number of subjects that you need in clinical trial. For example, to detect 50% of change in these markers, you need less than 50 subjects, while you need at least 100 if you use a clinical progression like the Unified MSA rating scale. Let's talk about functional PET and SPECT. Of course, I don't have the time to go into detail with this. What I just want to indicate is that with PET, you have higher resolution compared to SPECT, but definitely it's more expensive. It's not used in clinical practice, in clinical settings. So the DAT scan is generally more used when we want to look at the nigrostriatal system, which is the main target, the first target that we look at in people who come to our clinics with Parkinsonism. Both PET and SPECT in this case look at the dysfunction in the nigrostriatal system, and what you look in these images, this is the terminals, the dopaminergic terminals in the striatum, in the putamen, in the caudate. And that's what the images look like. I'm sure you're all familiar with that. And you can see that fluorodopa that looks at dopamine synthesis and storage, and the DAT scan that look at the dopamine transporter provide the same kind of information. There is a reduction of uptake in the striatum, particularly in the posterior part of the putamen. This is quite important for differentiate essential tremor and non-degenerative Parkinsonisms from neurodegenerative Parkinsonism, including multiple system atrophy. But if you ask if we can use these techniques to differentiate Parkinson from MSA, unfortunately, the answer is no. If you manage to match patients with Parkinson and MSA at a very early stage, similar symptoms, you see in MSA a more homogeneous reduction compared to Parkinson's, but there is a huge overlap. So that scan is not really useful to differentiate the two conditions. There is, however, another different type of PET scan with FDG, fluorodeoxyglucose. With this system, we can look at areas of increased or decreased metabolism in the brain. That is more useful. This is a patient with Parkinson, usually has a preserved or even an increased glucose metabolism in the striatum, while the patient with MSA has significantly reduced glucose metabolism in this area, suggesting that there is a reduction of function in these areas. There are softwares that enable now to look at metabolic networks in the different brain. And you can see that is definitely more important in Parkinson. You have increased in the thalamus and the anterior cingulate and reduction in the parietal area, while in the cerebellar you have, in MSA, you have a reduction of the cerebellar. And the FDG can also differentiate MSA from PSP and CBD, where you see that there is in PSP reduced metabolism in the midbrain, and here in the cortex in the SBD. It's not useful for the progression because, as I said, there is already reduction, significant reduction, so there is the floor effect. More recently, there are tracers for misfolded protein. We have good now for tau. For alpha-synuclein, we still don't have it, good specific tracers, but there are promising results in these studies. They have used this specific PET ligand that gives a signal in the white cerebellar white matter, in the cerebellar white matter, and in the medium peduncle cerebellar pontine, so it's able to differentiate MSA-C from DLB and Parkinson's. And you can also look with respect to the cardiac sympathetic innervation, which is normal is reduced in Parkinson's, but is normal in MSA patients. Although at the very early stages, even patients with Parkinson can have a normal signal. The conclusion of the first part, unfortunately, I'm a little bit behind. We still don't have a good marker, imaging marker for MSA. MRI is promising, and FDG also can be useful, but has not been standardized in centers. And more research is due for the new misfolding tracer protein ligand. And now, Jacopo will talk briefly about what we are doing in Newcastle.
Okay, thank you, Nicola. I'll try to keep it within the time. So what we did in Newcastle was using advanced MRI techniques and FDG PET to try and improve the standard MRI and techniques that we have at the moment. So going really to the interesting part is we tried to investigate microstructural abnormalities through diffusion MRI and neuromelanin sensitive MRI and coupling that with PET FDG. We had 11 early MSA participants within 3 years of diagnosis, and we compared them against 18 PD patients and 18 healthy controls, and we performed a range of clinical assessments. I've already gone into this, and I will go very, very fast on the methods, and if anyone wants to know more about this, I'll be happy to answer later. Neuromelanin is a pigment that stays in monoaminergic nuclei, substantia nigra and locus coeruleus. We can measure this pigment because it's hyperintense in certain MRI sequences. This was a manual segmentation and based on a previous investigation in PD. And basically, we can measure how many hyperintense voxels pertaining to dopaminergic neurons are left in the substantia nigra. And you can really see that PD and MSA have great reductions compared to healthy subjects. A similar concept applies to locus coeruleus. This was an automatic sampling of the locus coeruleus here at the very bottom near the fourth ventricle. And we measured here the hyperintense voxels as well. And you get a neuromelanin specific signal here and in the substantia nigra. With NODI, which is a diffusion MRI technique that allows to investigate, it's quite interesting compared to diffusion tensor imaging because it allows you to have both information about, more information about white matter and gray matter. And I'll go into this a bit later. It was an ROI analysis. I have to skip this. So locus coeruleus is quite interesting in MSA because it has been described, but not in this detail. Usually alpha-synucleinopathies have lower neuromelanin signal in the caudal parts and in the middle part of the locus coeruleus, while Alzheimer's disease is the opposite gradient. And you can see that in MSA actually, and this matches the neuropathology data, the scarce neuropathology data that we have. What was interesting is that locus coeruleus has been described, preserved neuromelanin in the locus coeruleus has been associated with cognitive function in elderly, healthy populations. That was true also for PD, and we know that, but we were not sure about that in MSA. And actually, we did find associations with MSA. You see both MSA and PD here, but these associations are significant in MSA, meaning that probably this is a physiological mechanism that goes through cohorts, and it's probably there as a basic mechanism in humans, I believe. We thought, why, if we had FDG PET data, if there was a connection between cognition and locus coeruleus and FDG PET, and actually neuromelanin content, neuromelanin signal in the locus coeruleus is associated with certain cortical areas, especially frontal areas, which also makes a bit of sense with the executive dysfunction that usually MSA, people with MSA have. With the NODI, we try to step it a little bit further than diffusion tensor imaging. And you can really see that free water that is in a parameter that implies neurodegeneration, neuroinflammation, how high it is in the putamen and in the cerebella gray matter, but also in other basal ganglia and in the substantia nigra. And finally, the intracellular volume fraction is still a parameter from NODI, and it tells you how packed axons are in white matter bundles, and you can really see that you can tell a difference between MSA participants in the middle cerebellar peduncle, cerebellar white matter, and the pons, between MSA and PD in the green, and healthy controls in the blue. We hope that we can bring this in the clinic, especially for clinical research and clinical trials, because we really need something to measure in the clinical trials, in disease-modifying clinical trials, other than symptoms and scores. Thank you for listening, and thank you to the Multiple System Atrophy Grant and to have enabled this research.
And thanks very much to both our speakers there for a great overview of imaging in MSA. So I'd like to now invite the final speaker to come and speak to us. So this is Professor Anya Lowett, who is based at the University of Strathclyde. She's an academic speech and language therapist whose particular interest is in motor speech disorders. And she's been the recipient of a grant from the MSA Trust to fund a novel programme to try and improve speech problems in people with MSA-C. So thank you very much.
Thanks very much, Chris. And I'm afraid this is a bit like Monty Python's, and now for something completely different. I'm not going to wow you with fantastic pictures of the brain, but it's going to be much more sort of a behavioural approach. And right, I'm not quite sure what's happened here. Oh, sorry, yeah, this is the introductory slide. The title is different. This is my actual title because I thought I'd go a little bit broader than the trial. I suddenly had a panic. This was in my slide. Anyway. What is communication? We all just sort of immediately think of speech, but it's actually much more complex. As this model shows, it's not just the speaker, it's to do with the listener. Obviously, speech and voice are very important for communication. But lots of other things are said as well. Language, can you find the right words? Can you understand what other people are saying to you? Very closely related to cognition, which we know is a problem in MSA. But also, you know, fatigue has a massive impact on the ability to communicate. In which environment are they communicating? Is there noise levels, etc.? How good is the listener in terms of understanding them and willingness to understand them? And also their personal factors, their own confidence, whether a talkative person before or not. So lots of different aspects to consider when we're assessing someone, but also plenty of opportunities where we can target our treatment. So just a little bit sort of quick overview of where is the communication breakdown. It is a very early symptom in MSA. And obviously we do have the MSA-P and MSA-C. They do have slightly different presentations, but there's also quite a lot of overlap, slowness of speech, definitely. But then obviously with the atax cerebellar side, we do these sort of ataxic movements with, you know, sort of prolonged phonemes and everything. So the rhythm out, whereas with the Parkinson's, we see the sort of very quiet voice, articulatory decay over time, very monotonous speech. There is, as I said, additional problems with language, particularly word finding, as well as the sort of general cognitive aspects. And what we have also seen across studies that have compared MSA patients with other related types like Parkinson's is that the speech impairment seems to be more pronounced than you would see in other conditions. Obviously, the speech mechanism is impaired, therefore you also have swallowing problems. It's nothing I'm going to talk about today, but just wanted to mention it because it's obviously a very important consideration in treatment. Just to quickly kind of show how patients perceive that. So we've run a survey recently looking at people with Parkinson's, MS, progressive ataxias, and people with MSA, and just asked them about the prevalence of their symptoms. So do they experience them often or not? And we can see the yellow bars there, which are the MSA patients, certainly kind of providing sort of indicators of highest prevalence across the board, cognition, articulation, the language that I was talking about, and also reporting highest assessed impact of these problems on their motivation to communicate, and also their confidence in communicating. Now, we might say they were just more severe, and that is true. So we did a sort of very rough indicator of their motor severity, and we can see that the yellow bars again are kind of more in that sort of higher severity continuum, but so are the grey columns, the progressive ataxias, and if we then go back to the other graphs, we can see that the impact there was less. So the, people with MSA definitely have greater problems with communication and therefore maybe in greater need for intervention. And we had sort of free text and says the impact that communication are having on these people. We can see in quite a substantive psychosocial impact of the problems, and people just feeling they don't get listened to, they feel isolated because of that, their confidence is down, there's embarrassment, there's sadness around it. So again, when we're looking at it clinically, we're very much looking at the whole person, taking that holistic approach rather than purely looking at their speech problems. Having said that, I'm going to focus more on speech now. So really there's been a massive dearth in studies, clinical trials involving people with MSA until about 5-6 years ago. All of a sudden we have a bit of an explosion. If I can name it, there's only really 4 studies, but there was nothing before. Because of that lack of evidence, therapists were quite often, what the feedback we're getting from therapists, they're not quite sure what is the best technique to treat people, and certainly when we've been asking the patients in our trial in terms of what SLT they've had, there's been massive variation there, both in terms of waiting time, some waiting very quickly because of their disorder, and others having to wait long. We've had people in the trial with quite substantive speech problems who have not seen a therapist yet, and amount of input varies massively as well as the focus and the nature of follow-up, which is, for me, the most worrying thing, is where some people are being followed up fairly regularly, and others are being discharged saying, come back when you need to, and they don't really know when they need to come back. So that is a big issue as well. So going to those studies, I said there's 4 that have been done specifically with MSA. They're all MSA-C studies, which I couldn't quite understand, but they're all Asian studies. And from what we heard this morning, this is probably why. So there's been a range of techniques employed. I don't know how many of you are familiar with LSVT. LSVT loud is a technique developed for Parkinson's, which just focuses on producing a loud voice that is then supposed to carry over into better speech. The Speak Out program is very much the same. So both of these have had some positive outcomes with people. They're very intensive programs, 4 sessions a week over 4 weeks. There's been water-resistant therapy as a voice therapy, but that seems to have also had some beneficial effects. The last one, the Sonora one, that's a Japanese study. They had people in hospital for 4 weeks, and they gave them OT, PT, and SLT each day of that. So it was very, very intensive. I don't know how these people dealt with that. And they had very little outcome, I'm afraid. They're really only sort of very physiological measures like breath support and oral diadochokinesis, and no sort of a little bit of a trend in terms of psychosocial well-being. Because we're looking at sort of parallels to Parkinson's and also ataxia, we can look at what's happened there. And again, we've had a big increase in studies in those fields as well. So Parkinson's, the LSVT loud, and the Speak Out, as I've mentioned. Singing has become quite popular in Parkinson's. There's no conclusive evidence for that, but some people have found some improvements. And then also in the ataxia area, a colleague of mine in Australia, Adam Vogel, he developed a kind of home practice app that takes people through exercises and give feedback, and with therapists check-in. And he had some good results there. And then I've tried LSVT with progressive ataxias, and also a sort of different version, LSVT Artic, that focuses more on articulation. So we're kind of starting to see evidence of different therapy techniques, and all of them result in some improvement. Thanks. So the one I really wanted to talk about was the most recent study I've done. Again, developed it with progressive ataxias first. It's a kind of combined individual group approach where the patient has 4 sessions individually to establish good speaking strategies, and then they go into a group with other patients to practice them over 4 weeks, and that's the intensive part. They meet every day. The kind of unique bit about this one is without the therapist, and so it's not a lot of load on the therapist, which has always been a big problem with the LSVT, where it's difficult to fit into their workload. We're kind of focusing on loud as well as clear in this, and it's done online so far. So we've had some really good feedback. I haven't got time to go through all this, but there's a general, social support, but also in terms of speech benefits with the patients leading the sessions. It kind of redevelops the agency. They can see other people using strategies and the effect it has, and it motivates them more. So we've had some good effects, and therefore we wanted to try that with people with MSA. So the current trial is this Clear Speech Together, and it's the first time anyone's actually comparing it to a different type of therapy. So we are doing what we call NHS standard therapy, which is 6 sessions individually over 6 weeks. And we've got 24, we're aiming for 24 patients. We're kind of looking at various aspects of their performance, both from physiological all the way to psychosocial levels. So we've got 23 recruited so far, 14 have completed everything. And you can see they're kind of relatively similar in terms of their motor levels. So far, the NHS group has got slightly higher intelligibility levels at entry. But in terms of outcomes, we're looking at roughly about 50% of each group having improved after therapy in terms of intelligibility as well as the naturalness. It depends a little bit on the timing of the assessment, but I haven't got time to go into that. Happy to explain that later. And the voice samples don't work. I'm afraid they're too quiet.
When I saw how I adjusted the rose into the bright sky, I knew it was for me.
Have to play it, otherwise I can't go. There we go. The other nice thing that we've seen is that despite not everybody improving in intelligibility, they have pretty much all improved in terms of their confidence and also their communication participation. So that's the blue to the golden line. And the other interesting thing, and this is only based on 50% of the sample, is that we can see with my 2-month follow-up, the people who are in the group are actually staying at the same level, whereas the ones who had the individual therapy are dropping again. So we'll have to obviously see whether that is maintained with the larger group, and it is still only a pilot, so we can't say for definite, but it's an interesting observation. So yeah, qualitatively, everybody agreed that the program met their needs and that they benefited, and family and friends are saying the same thing. Again, the other interesting thing, pretty much everybody said after the 2 months in the follow-up that they had gone worse. And that is coming through with the intelligibility measures. However, they kind of felt that if they hadn't had the therapy, it would have gone even worse. But obviously, we can't corroborate that with any measures. So very quickly, because we all want to go for lunch. What we have seeing is that people with MSAC can benefit from speech therapy, even if they're quite late on. And so there is really no argument of saying, you're going to progress very quickly, or you're too far down the line of not providing proper intensive, and just sort of send to people, just do voice banking and we'll get you an AAC device. The other thing is that even if there isn't much change in intelligibility, we can improve their confidence in their communication participation, which from a practical point of view is the important thing. It doesn't matter what the intelligibility is like, as long as they actually want to communicate with other people. They definitely need a much greater level of support and more regular follow-up, so that's very important to take into account clinically, so that their needs are properly met. And that is the end of my talk. So many thanks to the MSA Trust for funding the study, and to everybody else.
Thanks very much, Anya, for that great overview of communication problems in MSA. So I'd just like to invite the speakers to just come and have a seat here. And we've got time for, I think, a couple of questions before lunch. So does anyone want to start? Boyd Ghosh, do you want to make a start?
Thank you. That was really good. I just wanted to ask Professor Lowett. I guess you work in the NHS as well, do you, as well as academic?
No.
Okay. So I was just wondering how you might see translating your findings into changing practice for NHS speech language therapists, and what would be needed to to affect that?
Yes, so we've been talking with therapists all along to see if that could actually be feasibly built into the project. Some are sort of quite open to it, others kind of think that would be quite difficult to fit into current practice. The big problem we have at the moment is that if you're doing something with, say, MSA or even the progressive ataxias, they are rare disorders and you never have enough people in the one trust to form the group, but the project's been designed and the program is designed in such a way that it should apply to other types of speech disorders as well. So we're kind of hoping to be able to when we're going for a bigger grant, we would widen that out. But yeah, I mean, the therapists are able to make time for LSVT, and they are able to make time. The group studies are already quite prolific in the NHS, so it's just a sort of slightly different model, which hopefully shouldn't be too difficult to implement. But yeah, people need to kind of be aware of the, and the other thing is the technical support. So this is an online study. We wouldn't bring people into clinic every single day to do that. So it is, there's sort of additional support necessary with the online nature of it if they wanted to run that. Yeah. Thanks.
Okay, thank you. Do we have any other questions? I guess one point. One point that I wanted to make is, again, it's to do with resources in terms of, you know, the fact that patients in general are often discharged from therapists after one cycle of treatment, and I don't know if we need to improve that across the board, because I think that implies to other therapists as well as speech and language therapy, but it's a big problem. Okay. Do we have any other questions before lunch? Do you want to?
Hi, thanks. Katie Mayer from Ionis. I had a question about the standard MRI in terms of biomarker for clinical trials. Could you elaborate on which markers for a standard MRI may be helpful?
There is the study that I've presented for from the Prospect UK study. They have looked at different measures. And the best one seems to be the PONS. There is also the cerebellar white matter. The putamen can be used. And also the frontal lobe and the parietal lobe can be used. All of this, you need, talking about calculation samples, the number of patients in this progression intervention group could be less than 50, so that could help. For the atrophy itself, in other studies, we don't know. For example, based on the Innsbruck study, the PONS could be better in people with MSAC than PSP than MSAP. So.
I guess another point to raise, Nicola, is that in some studies, some recent trials have required certain imaging changes to be present at baseline to try and enrich the sample and ensure that we were recruiting appropriate patients, isn't it? And I think a question I'd like to ask you actually about the imaging component of the MDS diagnostic criteria for MSA. Part of that is diffusion-weighted imaging changes within the putamen and the middle cerebellar peduncle, and we don't routinely get those read out by radiologists in reports. So what do you think the implications are for potentially trying to implement that into more routine practice?
The one thing that we need to consider is that, unfortunately, all this MRI technique softwares to analyze are not being completely standardized across centers. So I think, and the same thing is also for FDG, that is very useful, but is generally not used just because there is no consent on how to. So I think the first step would be to standardize these methods, and that probably will be more widely accepted.
Yeah, no, I think that's a great point. Okay, well, I think if there aren't any further questions, I think we need to call this session to a close. So I'd just like to thank all the speakers again for great talks. So thank you.
So good afternoon, everyone. It's my great pleasure to introduce this afternoon session. So we're now going to be moving on to more clinical challenges in early MSA and diagnosis, which is what we grapple with in the clinic after a really wonderful basic science morning. So we're going to be focusing on diagnosing MSA in the early disease stages. So I'd like to hand over now to Dr. Wendi Phillips, who's a consultant neurologist at Addenbrooke's, where she's been doing an atypical clinic, a Parkinson's clinic, and a functional movement disorders clinic. And she's going to be talking to us on difficulties in the diagnosis of prodromal MSA. So over to you, Wendi.
So thank you very much for inviting me. I'm delighted to be here. So I'm going to present 3 cases. They've all got the same diagnosis that could have fitted into the possible prodromal category. As a reminder, that's urogenital failure, neurogenic orthostatic hypotension, or REM sleep behaviour disturbance, and subtle motor features. You don't need supportive features for this category, and you don't need radiology. So the first case is a 74-year-old lady who was seen in the general clinic in 2017. So she described REM sleep behaviour disturbance, unsteadiness and falls, mild memory problems. She's putting her clothes on back to front, and she's got a predominantly unilateral tremor. Here's her past history. So I've taken this verbatim from the letter. So testing her eye movements was challenging because she tended to shut her eyes. Some restriction with nystagmus and slowed saccades. The right upper limb looked apraxic, and her walking was markedly unsteady. So presumably because of the description of REM sleep behaviour disturbance and these motor features, the diagnosis of MSA was considered. An MRI scan was normal, so the DAT was sort of borderline, reported as borderline. So at review, this is a different neurologist, found that she had marked unsteadiness. She was now in a wheelchair, still described REM sleep behaviour disturbance, and she's developed bladder and bulbar problems. So no postural drop. Again, problem with these eye movements. Slightly hypermetric horizontal saccades. Voice hyperphonic. Foot tapping slow bilaterally. Mild finger to nose ataxia bilaterally. Slight tremor, and gait very unsteady and broad-based. So this neurologist felt she had MSA and commenced levodopa, to which she made a good response. On this review, she had, again, difficulty in performing eye movements, and her speech was very abnormal, but not typical for MSA. Her tremor was very irregular. It entrained to the frequency of a voluntary movement of the other hand. It was distractible with a complex movement of the other hand, and it paused with a ballistic movement of the other hand. She had very slow movements, but the movements were very slow and irregular, and there was no decrement. She was generally hyporeflexic, and she walked with a kind of robotic gait, like that. No postural drop. She had giveaway weakness and strongly positive Hoover's sign. That's a distractible leg weakness. So I discussed the diagnosis of functional neurological disorder, or FND. And testament to my communication skills, the physios reported that she was not invested in the FND diagnosis. So she was using visual reliance for balance, perhaps explaining the robotic gait, perhaps compounded by macular degeneration. She was seen by rheumatology because she had widespread pain, and they diagnosed osteoarthritis and pseudo-gout. She had normal urological investigations, but she told me she had a post-void residual, but the investigations were done in a different hospital. She was referred to the autonomic team at Queen's Square, and they thought over the phone she had a synucleinopathy, but they weren't able to see her because it was COVID. The gastroenterologist nearly put a peg in because they thought she had MSA, and a sleep study showed restless legs syndrome only. So she had no mental health issues, and she had muscle tension dysphonia, which is kind of functional dysphonia. So on review, she's on a huge amount of medications, still lots of autonomic symptoms. Repeat DAT was normal, but now she's still disabled, pretty much the same, but at least she knows she's now not got MSA. The second case, I don't have time to go over them in detail, but this is a 50-year-old man who, again, presented with lots of suggestive symptoms. He also had a borderline DAT. The consensus was that he had MSA. As time went by, it became increasingly clear that he didn't. And 8 years later, it was clear then that he had FND. And 8 years on, it was quite difficult for him to hear the change in diagnosis. And it transpires, in fact, that he's transgender, and he felt that that's what precipitated the FND in the first place. The third case is a lady in her 50s with a rapidly progressive MSA-like syndrome, some autonomic features, nothing structural on examination, but again, lots of these functional signs. But all she was interested in was palliative care. So I'm just going to spend the next few slides just talking a little bit about what a functional disorder is. So this is the formal definition. You need to have symptoms of altered motor or sensory function. You need to have positive clinical findings, so that's signs on examination. So in other words, it's not a diagnosis of exclusion. These signs are based on normalization of function with distraction of attention, or predictions about illness. And it's not better explained by something else, and it causes distress or impairment. So there's been a lot of interest in the pathophysiology of FND in recent decades, and several themes have emerged. So there's abnormal predictions about illness and increased attention, and that really reflects the signs on examination. Reduced agency, which kind of reflects the involuntariness of the disorder. Altered interoception, kind of limbic motor fast track, and impairments in planning and control, execution of movements, and awareness of intention to move. So FND is thought to be a disorder of the brain's predictive processing, and this is a theory put forward by Mark Edwards and colleagues. So the brain normally works as an inference machine to allow the brain and the body to be more efficient. So the brain makes predictions based on prior beliefs and expectations, in turn based on a lifetime of accrued experiences, based on family, friends, society, media, overlearned movements. And these predictions are sense-checked with incoming sensory information from the environment and the body. They're compared at multiple hierarchical levels in the brain and adjusted slightly, and eventually a perception is produced. So here's an example of the brain working as an inference machine. So this is a visual illusion. It's Einstein's face, but it's a hollow mask of Einstein's face. So hands up in the audience who can see that as a hollow mask. And so I'll demonstrate to you that it's hollow. There you go. And now this is the convex side. So I'm going to show you that it's hollow again. And now it pops up. So you know it's hollow. So hands up who can see that it's hollow now. Can you? Okay. So we're not supposed to be able to see it's hollow because we've got a strong prior belief that faces are not hollow, and that's due to our experience of never having seen people wandering around with a hollow face. And that outweighs the reality of our visual information. Similarly, in FND, patients have this rogue intermediate prior belief, essentially an abnormal illness belief, which gets stronger and stronger with attention, so largely subconscious attention. And there's less reliance placed on the sense-checking information from the reality. So in FND, there's a strong prior belief, strengthened by the attention, that there's a problem with our body, and this outweighs reality. So this is saying the same sort of thing. So you get a rogue prediction depicted by this gremlin, more attention paid to the abnormal prior, less attention paid to the environment. So there's a really big mismatch between the two. So you can't adjust and reduce the prediction error. This leads to reduced agency. So your brain's predicting something that's not sense-checked, which is why it feels involuntary. So what about functional Parkinsonism? So this is quite common. And a reminder, it's based on normalization of function with distraction of attention. A clue is that there's often a sudden onset or sudden change in the disease course. We talked about distractibility, entrainment, and pause with ballistic movement. Often the tremor in functional Parkinson's is in unusual locations, maybe here. Whack-a-mole is when you suppress a tremor in one place and it comes up somewhere else. And the rigidity and slowness can be distractible. We talked about the lack of decrement. So uneconomic postures, I'll have to come here and do this. So uneconomic postures is poor balance. but actually you need quite good balance to be able to do that. Walking on ice is a cautious gait, and robotic gait we talked about, that's kind of self-monitoring gait. Knee buckling, and the monoplegic dragging gait, it's like this. And that's a good example of how you would walk if you had a prediction about what it would be like to have a bad leg. Often with functional ataxia, you see a really exaggerated dysmetria out of proportion to other signs. So it's like a motor inconsistency. And difficulty with eye movements just probably reflects difficulty initiating voluntary movements. So it's really important to remember that FND very often coexists with or is triggered by structural disease, in particular Parkinson's. So you can get functional Parkinson's, which is just FND, and then you can get it comorbid with each other. So you've got to look at the whole picture. It's best to try and elicit multiple signs, and things that are bizarre doesn't mean FND. So there is a link between FND and Parkinson's. It's comorbid in about 7% of patients. And it may be because in both disorders, you're kind of stuck in this goal-directed self-monitoring mode. There's reduced self-agency in both disorders. And there's another theory that there's disinhibition of the default mode network in Parkinson's. And that's where a lot of these priors are thought to be generated. There's also a big placebo effect in Parkinson's, emotional dysregulation. And it may be just that the physical precipitant, so this idea of another disorder being a symptom scaffold for functional disorders. It's important to recognise, obviously, because it's very common, and the treatment is different. So MSA doesn't tend to be comorbid with FND, but as we've just seen in these cases, FND can present as MSA, probably just because FND is very common, particularly in younger patients. And if neurologists have got less experience with MSA, they might tend to think, well, if you've got odd-looking Parkinsonism, that might be MSA rather than Parkinson's disease. Autonomic symptoms are common in FND, as is poor balance, slowness, and difficulty with eye movements. It could be mistaken for MSA. And functional Parkinsonism may respond to medication, making you think you're on the right track. So in conclusion, just a few learning points, particularly from the first case. Obviously, it's much easier in retrospect and with the passage of time to make these diagnoses, but it is difficult to undo a diagnosis once it's been made. And with all these patients, they presented with lots of the right symptoms, but it's important to have objective verification, particularly with the REM sleep behaviour disturbance. And the REM sleep behaviour disturbance in the criteria is actually polysomnography proven. The data is often helpful, but as in those first two cases, it can sometimes confuse the situation. Always remember that comorbidity can trigger off FND. Just in terms of logistics, you've got to make sure you get all the information. And we've talked about the link between Parkinson's and FND, important to recognise because the treatment's very different. Thank you.
Thank you. So thank you very much for a very interesting talk that I'm sure will generate a lot of discussion, Wendi. But we will be moving on for now to the second talk. And this is going to be given by Dr. Yen Go, who is an experienced neurology trainee doing a PhD. And she's going to be looking at clinical progression markers in early MSA. Over to you.
So essentially, over the next 15 minutes, I am going to talk, don't worry about the, if we can't see it, we can't see it. I'll wing it.
Wing it?
Yeah.
Sure. I don't have to.
Okay. I'm going to talk about our current understanding of clinical prognostic markers in MSA, and then spend most of my time talking about results from the Queen Square Brain Bank prospect, sort of like deep phenotyping work around this that I'm doing, and then maybe finish up with the bit that I actually think is really interesting, and I'd quite like to hear other people's thoughts on it, comparison with other international cohorts and the implications of that. So, this is stuff that everyone in this room probably knows. Survival and progression in MSA. So mean or median survival is reported differently across different studies, but it's usually between about 6.9 to 10 years. But we all know that there's a benign subtype that exists where patients can live more than 15 years. And you've got, I think the longest reported neuropathologically confirmed patient lived up to 23 years. And for most of that, it was actually thought to be Parkinson's. Conversely, you've got other patients who deteriorate and die within 5 years. And we don't quite know why. And the main issue for that for us who are clinicians in the room really is that, when a patient comes in, you can't really tell them what's going to happen, how do you plan, how do you do any sort of advanced planning, but also for clinical trials. If you've got a very, very variable cohort, how do you even start to analyze your data? And that's kind of the question that we're asking. And this isn't really a new question. This slide really is just to kind of show you what other people have done in the past. There are many possible negative prognostic factors down the middle, but what you will notice is that for every study that has said that this is a possible negative prognostic factor, there is another study that said, actually, no, it isn't. And the reasons for this will be a bit varied. Part of it is going to be that actually, most of these studies aren't pathologically diagnosed studies. So there are only 4 studies on there in bold, which are neuropathologically confirmed postmortem studies. And the total cumulative number of patients in those is only about 200. Everything else is clinically diagnosed. And I think, as Prof Gentleman was saying earlier on, actually, you can get a misdiagnosis rate of up to 40% in some postmortem cohort studies. And so that really kind of messes with what we're doing. I'm not going to go through everything in great detail, but this is just an example of where we are now. Basically, we don't know. We think we've got some ideas, but we don't really know. So the aim of the work is to try to distinguish clinically accessible prognostic factors, and really ideally within the first 3 to 5 years of onset. And the reason for 3 to 5 years is because on average, your time to diagnosis is about 3 years. And so what you really want to do is be able to see a patient in clinic, and within the first visit, or maybe second or third visit, be able to tell them, well, actually, this is where I think you're going. Are we looking at 9 years? Are we looking at 15 years? And so this is the work that I started doing 2 years ago, which never seems to end. Essentially, we're looking at two different cohorts of patients. The first one is the Queen Square Brain Bank cohort, which is where I did a retrospective case note review of I'm not even going to try to say it, of real-time primary and secondary care documentation. This also included neuropathological reports and clinical summary data. And in total, there were 273 patients, of which 254 had enough clinical information for us to be able to do anything with. The second cohort, which I think a lot of people in the room will be familiar with, is the Prospect-M cohort. Now, this is a UK-based prospective cohort study of patients with atypical Parkinsonism that are followed up over 5 years with history, examination, clinically validated rating scales. And through this, we have 318 patients with clinical MSA identified, 21 of whom were confirmed on postmortem. Just so you know, when I talk about the cohorts later, 17 of them overlapped. And these are the cohort characteristics for anyone who's interested. Essentially, they're quite normal in terms of cohorts. We have a slight predominance of males, and I'm not quite sure why. We just have more men in our cohort of 555 people. The mean age of onset was 58 years, with the mean age of death being 66 years. The only things I would want to point out is between the Queen Square and the Prospect cohort, the Prospect cohort had, on average, a 2-year later age of onset. But survival was actually the same because they died a couple of years later as well. And I don't really know why yet, but overall, the actual survival period looked about the same. This is a histogram of all the surviving patients. And this is just to, again, kind of reinforce the idea that if you look at the survival group of less than 5 years, there's a 15.9% of all the patients fall into that group. And more than 12 years, 9.1%, which suggests that 25% don't typically fall into the pattern. You know, when patients come to you and they Google, how long am I going to live? 25%, 1 in 4 of your patients will not fall into that category. So this is probably the most boring bit of the results, the bits that didn't work. Okay, so survival is independent of both sex and clinical subtype in our cohort. It was also independent of onset symptom type. And this is probably a bit more important because there have been quite a few different studies that suggest that people with early autonomic signs or symptoms have faster progression, but that's not something that we saw in our 500-strong cohort. What we did see, which was somewhat interesting, was that in the post-mortem cohort, patients with an SND predominant sort of like, and I'll be honest with you, this is a subjective diagnosis. So the pathologist looks at it, they give a subjective diagnosis of whether or not they think this is more SND, more OPCA, or whether it's mixed. But the SND predominant cases had a shorter survival time, only by about a year or so, but you can see that it is quite clear there. Alongside that, what we also found was that younger patients survive longer. And so on the left-hand side, you've got a scatter plot that kind of just shows a very clear, fairly convincing sort of like best fit line. But also, if you look at the Turtle Kaplan-Meier plot on the right-hand side, it's quite clearly the younger you are, the longer you survive with MSA. And what was quite interesting was that actually the SND predominant patients did have a later age of onset, which makes it quite well, I say confusing. I suspect the SND finding that we have, it might be a confounder rather than anything else. And the reason why I say that is because I don't really know what happened to my slides. Okay. I had a next slide. I had a next slide that kind of compared SND and young age of onset. And actually what you find is that when you put them in a multivariable regression analysis, the effect of SND predominance actually disappears. And it's actually the age of onset that's a stronger predictor of survival rather than SND. Oh no, that is, sorry. It is there. That's the thing. And for the people interested in statistics, I did check interaction terms, I did check for collinearity, everything was non-significant, just in case. So the next thing we're going to look at, mobility and bulbar milestones. So these are some of the mobility milestones that I looked at, and across the board, early loss of mobility is associated with shorter survival. So if you have recurrent falls, walking aid use, kind of like, and all of that. One of the things that I thought the clinicians might find interesting is the median years of attaining any of the milestones. So the median time to having recurrent falls is 2.6 years. To using a walking stick, walking aid is 3.2 years. Outdoor wheelchair use 4.7 and indoor wheelchair use 5.7. And it kind of just gives you an idea of if that's the median, where does your patient fall within our cohorts. But as a clinician, I found the kind of hazard ratios and all of that kind of thing a bit hard to understand. Well, I find it hard to understand, but that's me. So what I've done is I've sort of translated that into Kaplan-Meier plots, where you can see that for falls and walking aids, there's not that much of a difference. So at your 10-year survival, if you fall before 2.6 years, is 25%. If you fall after 2.6 years, is 33%. And similarly for walking aids, it's statistically significant, but not clinically useful. It's a bit better with outdoor wheelchair stuff in that you have, with an outdoor wheelchair use of less than 4.75 years, you're looking at a 23% survival rate at 10 years, whereas it's 42% if you're not using a wheelchair around 5 years' time. And what was even stronger was indoor wheelchair use. So at 5.7 years, if you're using an indoor wheelchair, at 10 years, you only have a 10% chance of survival. Whereas if you aren't using a wheelchair by about 6 years, your 10-year chance of being alive is about 50%. And that's really quite stark. I won't go through all the numbers because there are lots of numbers on these slides. I'm very happy to share the slides with anyone who's interested, but it goes through the same thing. The thing that I would pick up on this one is the unintelligible speech. So if someone has any degree of unintelligible speech by 5 years' time, they actually have a 0%, in our cohort, they had a 0% chance of survival at 10 years, whereas it's a 49, which is nearly 50% chance at 10 years if you don't. Okay. And then we've got autonomic milestones. Again, it's a similar thing. The earlier you hit catheter use, the earlier you hit incontinence, and the earlier you hit needing blood pressure medications, the shorter your survival. Just going to go through this very quickly. But then what we did was that we combined everything into a multivariable regression analysis. And just in case, we only used variables that were available in both cohorts for numbers reasons. And what we found that actually, the important things to look at were indoor wheelchair use and unintelligible speech, whereas age of onset, time to falls, all of that just disappeared in the model. For dysautonomia, we had to do a second model just because it was only available in one of the cohorts and not both. And in that, what we found was time to catheter use and time to orthostatic medication use was the important thing, not urinary incontinence. So The next thing I'm going to kind of go through quite quickly is rating scales and survival within Prospect. So in Prospect, we looked at the UMSARS, which is an MSA-specific disease progression scale, the MSA-QOL, which is a quality of life scale, PDQ, which is a Parkinson's-related health quality scale, and GDS, which is a depression screening scale. And so this scatter plot is just to say your UMSARS goes up with time. These are all the UMSARS that have ever been done within the Prospect study. The longer your disease duration at the assessment of the UMSARS, the higher your UMSARS. And the mean disease duration at baseline for a Prospect study was about 4.7 years, and your median baseline UMSARS was 44. And what we found was that you have a higher baseline UMSARS was associated with shorter survival from baseline assessment, but not from onset in itself. So you can do an assessment UMSARS, and you can use that to predict from that point, but you can't use it to predict from when they started. Which kind of makes sense because the UMSARS is associated, with things like mobility and things like that. And the worse it is, the further on in your journey you are. But this is the bit that's probably quite interesting. So baseline UMSARS of more than 44 was associated with a, relatively significant difference in terms of survival. And looking at your survival point from doing that UMSARS, if your UMSARS is more than 44, then you actually have a five-year survival rate of 17%, whereas if your UMSARS is less than 44, then it's a 44%. So if anyone's got time in clinic to do UMSARS, please go ahead. Then the next thing we looked at was the rate of change of UMSARS. So 91 patients in the Prospect cohort had multiple longitudinal UMSARS scores. So we could calculate, using simple linear regression, a mean rate of change. Now, the mean rate of change per year was 10.47 with a median of 10.27. And here's the interesting thing. So your rate of change of UMSARS does not change with your disease duration. So for the Prospect cohort, this is the important thing to say. For the Prospect cohort, your rate of change of UMSARS did not correlate with disease duration. So it doesn't matter which part of the disease duration you're at, it didn't actually make a difference. And a higher rate of UMSARS change was associated with shorter survival, which I don't think is a surprise to most people. But using the Kaplan-Meier plots again, so if you do your UMSARS, and then a year later you do the second UMSARS, and there's a more than 10.2 point change in it, then what you're looking at is a 5-year survival rate of 16%, versus if you've got a less than 10-point change in your UMSARS, you're looking at a 5-year survival rate of about 50%. And that could be quite significant for some patients, given that typical disease length is only like 7 to 10 years. And very quickly, we did a few other rating scales. They weren't quite as interesting, but if you do a baseline MSA-QOL, that can be used to predict a little bit, but it's not very strong. Whereas your PDQ and your GDS did not have any association with that. Now, this is the bit that I actually find quite interesting. So you can see at the bottom line, these are the rates, these are the median survival from onset rates for the different studies. And what I've got up on the system is that I've got the Queen Square Brain Bank cohort, the Prospect cohort, and then I've got the 3 prospective MSA cohort trials. And these are, the reason why I've put them up and not the rest is because they are the actual natural history prospective trials that everyone looks at. And what's interesting about it is that in the UK, our survival rate is starkly different from theirs. And the question is why? So I kind of went, is it real? I looked at the diagnostic criteria. Everyone uses the Gentleman criteria. And according to the papers anyway, there shouldn't be any difference in diagnosis criteria. Then I looked at age of onset, and you can see the age of onset there actually doesn't make a difference. The French reference center was a bit older than us. The North American center was much older than us. It's not the age of onset either. If you look at, say, UMSARS progression, again, if you look at the Wendi paper from the MSA cohort, actually your UMSARS progression over the first 2 years is much higher than what we're seeing in the Prospect cohort. And so it is a bit of a mystery. I've got some theories as to why it may be happening. It's something to discuss. That those cohorts have obviously been published. We are still collecting and ongoing. Did COVID have a result in it? We can't really know. Looking from a year-to-year basis, there's not been a spike in deaths, however, so I can't really say that it's going to be COVID. Is it a difference in cultural attitudes towards life and death or treatment of a palliative condition? Again, difficult to know. Or could it just be healthcare infrastructure? Because we've got very different healthcare infrastructure here. And if it is healthcare infrastructure, then it's certainly something that needs to be discussed. So, in summary, the formatting has really enjoyed itself there. There are some predictors of survival from disease onset. The main ones being time to wheelchair use, unintelligible speech, catheter use, and how soon you need your BP meds. Age of onset is associated, but it's weak. And then you can also use some predictors of survival from assessment point, which include the UMSARS and the rate of change of UMSARS, as well as the MSA-QOL. And I think the formatting was actually just reading my mind, because that is actually the most interesting to me, the most interesting thing to me right now. Why is survival in our UK cohort lower than equivalent European or North American cohorts? And I look forward to the discussion.
Brilliant. So it's my great pleasure to introduce Professor Kailash Bhatia, who's a neurologist, professor at Queen Square here, and who has always given fantastic teaching and training. So we look forward to hearing your insights, particularly on diagnostic dilemmas in early MSA.
Thank you very much. I think my talk sort of follows up nicely from what this morning Ulrika presented about the difficulties we have in many of the cases. And she concentrated on the ataxia side, but I'll be sort of thinking about it in a little more broadly. I'm not going to sit and give you the diagnostic criteria. You heard about that at least twice today. Suffice to say that the criteria are well established, and we now recognize the supportive criteria quite well. The core features of levodopa refractory Parkinsonism, cerebral ataxia, autonomic, are shared with many other disorders, which may have different pathologies. And in this context, already we've been alluded to these two studies where the diagnostic accuracy, even nowadays, if you say contemporarily, 62 to 79%. So 20 to 25% rate of an error. And this one in the morning we were talking about this, and here's another one from the Queen Square group itself. But both of these have actually concentrated on DLB, PD, and PSP. And again, I'm not going to go into this because we do know about how to think about differentiating these disorders, early onset autonomic, early onset dysarthria, stridor, etc., making it more likely to be MSA. What I want to talk about, however, is some other scenarios which I want to sort of draw attention to. So there are two sort of characteristics here. Examples of MSA which may be atypical, hence not considered. So amongst them is young onset MSA. We haven't heard about that today. And sometimes very asymmetric MSA, which looks like CBS to some people. And then on the other hand, there are conditions where we think this is MSA, but they are actually due to other etiologies. So what you may call MSA mimics. So young onset MSA, we heard just now, in most of the prospect study, etc., that the average age of onset is around 58 to 60. And what I find then is that people who come with the young onset variety, and just like you have in Parkinson's disease, the young onset variety, there is also the young onset variety of MSA. And a number of these people are not picked up early on, even because we just don't think about it. We think it's a genetic form of Parkinson's. We think it's something else going on. But these people have young onset MSA, and half of this cohort was pathologically proven. So we're not just diagnosing them clinically. I just want to show you an example. Now, this lady, you'll hear her, or you may not be able to hear her. She presented with what was thought to be a vocal cord paralysis. She went to the ENT people first. She had stridor. She was very young at that time. She was around, I think, 38 or so. And
1, 2, 3, 4.
You can hear her speech is affected. She went through from one ENT person to another ENT person. Nobody thought about looking at her. If you see her walking, you'll see straight away that she's Parkinsonian.
3 years ago?
We go around here.
Are you using a walking
None of the people
aid?
pick this
Sometimes no.
And eventually when she comes to us with that thing, with the stridor, with the vocal cord paralysis, etc. This is young onset MSA, went on quite rapidly in 3 or 4 years. You'll see a jerky tremor of her hand with a myoclonus which was present. She had seen at least, by this time when she had developed the Parkinson's, 4 different neurologists. Nobody thought about young onset MSA because we just don't think about it. It's an unusual sort of a condition. You can see the jerky tremor very clearly.
Close your eyes and count backwards from 100.
So the other one, again, is a asymmetric presentation of multiple system atrophy. We normally think about MSA as a symmetrical disorder, but there are from time to time people who present to you who are asymmetric. And again, in this cohort, I think almost half of them have pathology. So this is a gentleman, unfortunately we didn't have a video of him right at the beginning when he was very asymmetric, but this was the arm which was more affected, and you can see the jerky tremor, you can see a bit of dystonic posturing, and so on. The other side is less affected, and he was thought to be CBS. Later on, he actually even had a little bit of slowing of his eye movements, which was unusual, but he did turn out to be MSA with all the characteristic features on imaging, unlike CBS. So that's another one I want you to keep in mind. The second group is what I call atypical Parkinsonism. When we first sent this paper to Movement Disorders, he returned it, said, there's a mistake in the title. It was Jose Obeso. And I said, well, the first one is in, as you can see, in inverted commas. There's no mistake. This is what we actually mean. And what we were talking about here are people who look like MSA or PSP for that matter, but they have certain features which may be atypical. Okay, and that's for you as clinicians to pick that up, and because there are a number of these cases who have a genetic condition underlying. So here's an example. This man comes to me with a 5-year history of tremor. He's 55. He's got Parkinsonism. He has imbalance. He has postural giddiness, mild cognitive dysfunction, no response to levodopa. The postural giddiness was documented to be postural hypotension.
Like this.
And at that time, for some reason, our MRI scans were very delayed. We had a waiting time of 9 months, but you could get a DAT scan in 2 weeks. And somebody did the DAT scan, and they reported to me it's normal. Now, this is a red flag. If you got Parkinsonism, there are sometimes people with MSA who might have a normal DAT scan. But if you got the Parkinsonian phenotype of MSA, your DAT scan should be abnormal. So you say, oh, what's going on here? This should be picked up as a red flag. And not surprisingly, there you see, once we did get the MRI scan, the penny drop. This is middle cerebellar peduncle, hyperintensity, and this guy had Fragile X premutation, 88 repeats, and he presented like a MSA phenotype. Here's another man, almost, you may say, almost classic presentation of MSA. He's in a wheelchair, he's got cerebellar signs, he's got pyramidal signs, he's Parkinsonian.
Okay. Close your eyes for a few seconds.
He's got a bit of myoclonus, a little bit of a peripheral neuropathy.
Big movements.
So you can see the bradykinesia here very clearly. You see that little bit of dystonic face also, because he's on levodopa, and you think of that in MSA. You can see the ataxia.
Okay, now with the left hand. Keep going.
Okay. Very slow. Brisk reflexes, so pyramidal features. That's the sign. So this is a tip for you.
For me.
And I always examine my patients with MSA or Parkinsonism. Remove their socks and shoes, because there is the clue. Okay? That's the clue. So this guy has CTX. Okay? Now we think about CTX as an early onset disorder with cataracts and this sort of thing. Number of papers coming out now that CTX can come with Parkinsonism. It can resemble MSA and sometimes can also resemble PSP and so on. So this is a treatable disorder in a sense. Okay. The other interesting thing about CTX is that actually it's a tauopathy. It resembles algorithmic brain disease. So it's not a synucleinopathy at all. And if you were to include this guy in your trials, then you'll be having problems. Okay. So MSA look-alike with regard etiologies, they are possible in all 4 domains. So when you're thinking about Parkinsonian forms, keep in mind Gaucher's disease, mitochondrial disorders, Fragile X, some of the spinocerebellar ataxias presenting only with Parkinsonism, as we heard about this morning. If you're thinking about autonomic domain, Fragile X, scars, CTX, if you're thinking about cerebellar forms, number of the scars, 1 to 6, 12, 27, we heard about that this morning, CTX canvas. And in canvas, your MSA patient with a cough, unexplained cough, somebody was coughing there, unexplained cough in an MSA patient, canvas. That's top of your list. Pyramidal signs, SPG11 and SPG7. And interestingly, the SPG, so in a MSA patient, you are looking out for that pyramidal feature. But if the pyramidal features are shouting at you, and you have clonus going on, and you got really marked as, no. That's not the phenotype of MSA. So you've got to think about that. And SPG7 and HSP7 may underlie a number of cases of what we used to call ILOCA. So these are people who are coming to you later as they are MSAC. So this is, I think, a very important, apart from the one which you showed this morning, Vioroka, the SCAR17. This is a very important cause of a MSA mimic. So this is a pathway which we sort of documented in that paper. You have an MSA phenotype, and you're thinking about that, what are those atypical features? What are atypical for MSA? Too young, for example. Although I showed you the young onset MSA, but if it's too young, then all of these things you have to keep in mind, CTX, mitochondrial, SPG11, and so on. Tempo of evolution, you heard about the tempo just now. But if it is too rapid, then prion, other things, and so on. Family history. Is there a family history? Normally, most of your patients with MSA don't have a family history. But if there is a family history, then some of the scars, Perry syndrome, etc. No family history or recessive, again, think about these disorders. So you can follow this little algorithm when you are faced with a patient where you think that there's something unusual about this patient with MSA, and you can use that pathway or algorithm to try and think about these conditions when you're thinking about that. The other thing I think I want to talk about, which has become very important, is that apart from the genetic causes, immune-mediated is become important, okay? And in that context, of course, IGLUN5, mainly because IGLUN5 has this REM and non-REM disorder. So when the patient comes to you and they say, do you have any problem in sleep? Yes, he's jumping about, he's doing things, he's doing this sort of finalistic movements. You think that that's RBD, okay? But these people have REM and non-REM disorder. IGLUN5 is on the interface between autoimmunity and neurodegeneration. The pathology is a 3 and 4 repeat tauopathy. But why is it important from the MSA point of view is that a lot of these people get dysautonomia. And they get dysautonomia quite early. A lot of them get stridor and they get sleep apnea. And they can present with ataxia. So you're seeing somebody with ataxia thinking that there is RBD and you have stridor and so on. And you must keep in mind IGLUN5. Okay? So the movement disorder can be PSP-like, but it can also be ataxia and so on in your this. Now this is a nice paper from Dalmau where they, as they mentioned, dysautonomia, stridor, etc. are important in MSA. They looked for, but also in IGLUN5 antibodies, but in a very small cohort of only 18 patients, they looked for it and they did not find a single case. However, since then, I've been to Spain a number of times and they have shown me at least 4 cases of MSA phenotype of IGLUN5 with the stridor, with that. And at this point, when this paper was written, it was said that this is not treatable. But with IVIG, in 3 of those 4 cases, they have responded and they are still surviving 5 or 6 or 8 years down the line. So I think this is an important thing to keep in mind. The other one is HOMA3 antibody. And this is again a potentially treatable cause. Looks like MSAC, can have autonomic dysfunction, etc. But mainly, and they respond again to IVIG, plasma exchange, and so on. So another cause to keep you, why it's important. So why is this recognition and phenotyping important? Well, disease-modifying, we're talking about disease-modifying therapies. But in 2022, all of these things, these were all negative. If you've got a rare disorder, and if your treatment effect is very small, if you're thinking about that, and if there's a contamination, even in a small number of cases, your treatment effect is, so really we have to be very careful about making the right diagnosis in these people. And hopefully we heard about this morning. We are hoping that this comes through, that we have the seeding assays and so on to be able to tell us whether the patients we are describing are really MSA of the synuclein variety or not. So a number of conditions are being recognized which imitate complicated forms of Parkinson or atypical Parkinson like MSA. A number of them are genetic. You already heard this morning about some of the genetic varieties. But I think Fragile X, scars for MSA, keep those in mind. I think where necessary, you're thinking about doing genetic panels, whole exome or NGS, and also immune conditions which may imitate or look like MSA. Thank you.
Thank you very much, Kailash. So, great pleasure to introduce Dr. Valeria Iodice, not quite, who is a neurology consultant. She's also an autonomic expert, and she's associate professor here running the unit. So we're very interested to hear your insights on the study.
Thank you so much. So good afternoon to all of you, and thank you for the invitation. Okay. So we have heard in several talks this morning that autonomic features are very frequent in all alpha-synucleinopathy. They can affect all the different autonomic domain, from cardiovascular with the neurogenic orthostatic hypotension, gastrointestinal dysfunction, sexual dysfunction, pseudomotor, and urinary incontinence. And you see the cluster of alpha-synucleinopathy. So I'm going to focus on, in particular, to the pure autonomic failure. So in a patient that presents just with isolated autonomic failure, degenerative in nature, and I'll give you some red flag later on, these are the patients defined as a pure autonomic failure. Now, the problem is then the clinical diagnosis of pure autonomic failure it can be made in a patient that then in the early stage of multiple system atrophy. So then they will develop later on actually different kind of alpha-synucleinopathy. And this has been confirmed in two autopsy-confirmed studies. So the first one on the top is from the Mayo Clinic cohort, so with Phillips, where actually a third of the patient, in fact, they presented just with an isolated autonomic failure. And we have a very similar results in the brain bank here, Queen Square, with a window of 1-6 years. So there is this premotor phase, which is also our window of opportunity in a way, where we can identify the patient in the very early stage, patient presented just with urogenital dysfunction, symptomatic OH, or REM behavior disorder, who unfortunately they do progress to such obviously an aggressive pathology. And to put everything in the context, I'm going to give you two examples of patients that frequently we would see in an autonomic clinic. So the first one is a patient with a pure autonomic failure. So you see on the very left side of the slide, this representation, where what we meant with that is then in a patient with a pure autonomic failure, then it doesn't evolve in any other alpha-synucleinopathy, you would expect, by definition, then actually there is an impairment in the peripheral autonomic nervous system, so ganglia and autonomic nerve. So this is a patient, 58, female, long-standing history of anhidrosis, neurogenic orthostatic hypotension, urinary frequency, severe constipation. We have seen this patient at 10 years of follow-up is not evolving in any other alpha-synucleinopathy, so it's appropriate to label these patients as a pure autonomic failure, and this is what we would see in an autonomic lab. So the patient presented, this is our thermoregulator sweat test, so the chamber, where we can measure the full map of sweating output. Unfortunately, this patient had a generalized anhidrosis, which is very much the case in many patients with a pure autonomic failure by the time we see the patient in clinic, with no doubt widespread cardiovascular autonomic failure. Now, second example, autonomic failure evolving in multiple system atrophy. So this is a patient, you will see a different representation here. So historically, in patients with multiple system atrophy, you would expect then when it comes to the autonomic nervous system, there is a central involvement of the autonomic nervous system. This is a 63-year-old man at the onset with anhidrosis, so lack of sweating, heat intolerance, feels exhausted post-meal with minimal activity, urinary frequency, urgency, incontinence, and nocturia up to 9 times per night. REM behavior disorder as well. So when we saw the patient the first time, that was the phenotype. So there was no neurological feature. We saw the patient in the lab, which is, you can see on the right side of this slide, and there is no overt orthostatic hypotension. Obviously, we do a full comprehensive autonomic assessment, and there is no activation of the baroreflex, so definitely there were emerging adrenergic dysfunction. When it comes to the pseudomotor side, so you see a different pattern you have seen in the previous patient. So definitely anhidrosis, but the distal part, actually the sweating is absolutely normal. When we use a different test, which is called the dynamic sweat test, we stimulate the postganglionic fiber, which are pretty much intact. So put everything in the context, this patient definitely that is not typical for a pure autonomic failure. So this is a patient with a central involvement of the autonomic nervous system. I'm not presenting here also the catecholamines were normal supine without any, with an increased upright. 6 years later, this patient developed Parkinsonian feature and was diagnosed with multiple system atrophy. Obviously, there is the huge challenge for the clinician, the autonomic neurologist in clinic, when you are without, obviously, any overt Parkinsonian feature, you would label this patient, you have no choice as a pure autonomic failure. Even though you have high suspicious in clinic, then that is not the case. So, as Yen has also discussed, so autonomic feature in the early stage of the disease are very frequent in all alpha-synucleinopathy in reality. Yeah? So it's not having or not having autonomic dysfunction, but it's the pattern. So when the autonomic dysfunction they do occur, how progressively they do develop, so how fast is the progression on the autonomic side, then it might give us some clue regarding which kind of a trajectory the patient will take. Now, I'll give you an example. So for the sake of the time, unfortunately, I can't present all the study, but just to give you an outline of how are the cohort, the longitudinal cohort worldwide. So a third of the message is, a third of the patient with isolated autonomic failure, they do evolve into another alpha-synucleinopathy. And you can see the numbers are pretty much similar. So two studies from US, so an important milestone with the natural history study led by Horatio Kaufman and the American consortium. You can see this is a prospective study. 34% of the patients, they did convert to another pathology within 4 years. What you see on the top is the cohort from Mayo Clinic with Wolfgang Singer. 48% they were the converter, and that is the other challenge. This is something that we need to, all of us, we need to agree on, what we define as a conversion. This is the cohort from the Bologna group. So 50 patients, 16 of them they did convert, so 32 converted to other alpha-synucleinopathy. And this is our retrospective and prospective Queen Square autonomic prodromal study. We have 283 patients with a pure autonomic failure, and we have a conversion rate of 36%. However, keep in mind that this 36%, this is with the new criteria of multiple system atrophy. So within this 36%, there are 9 patients already in the prodromal MSA. So to summarize so far, the story doesn't fit all the patients, so I don't think we can use we can't imagine that the phenotype in a patient evolving into a multiple system atrophy is progressive and is central, and patient evolving into Lewy body pathology is slow and peripheral, because there are many patients in the middle. So we have seen that actually they are quite heterogeneous in their biology. So it remains to answer many questions. So we need to identify biomarker in multifocal pathology. So which biomarker we use where actually a multiple system atrophy doesn't have just the involvement of the central autonomic nervous system. Our autonomic biomarker in the promotional phase, do they perform quite well? And do we have a biomarker actually to measure progression of the disorder? And this is what also Roy Freeman was raising as a question this morning. And this is where it comes our study. So you can see then it does have a retrospective, a prospective arm. The very first patient, we have a luxury here, obviously at Queen Square, because of the long history of the autonomic department, started by Sir Roger Bannister and expanded by Prof Matthias. So the first patient with a pure autonomic failure was actually assessed in 1973. In 1980, we did establish the plasma biobank. So we have a plasma sample for all these patients that we have characterized. And we did look at several studies. I'm going to just give you an outline of some of them. So our phenotype of the retrospective patient, 194 patient, this is the work of our PhD student, Dr. Kohai. So all patients, regardless if they are retrospective or prospective, they assessed we see the patient at least once a year with the biomarker that you see here. So patient will have cardiovascular, pupillometry, bladder function test, obviously the catecholamines. So within this cohort of 194, 26% are the phenoconvert. So patient then at the last follow-up, they have convert in other pathology. So you can see that it's very different from the phenoconversion rate if we are including a different criteria. So if we are including the prodromal, obviously we have many patients more phenoconverting. And these are very consistent also with the natural history from the American consortium. So having a noradrenaline higher than 200 picograms, an heart rate variability to the breathing more than 10, normal pupil, an orthostatic intolerance -6, which means a better orthostatic intolerance upright, is a risk factor for conversion to other pathology. We were also interested, and this is again the study of Juni Kohai, looking at the alpha-synuclein in the skin of this patient. So far, this is a prospective study, so we are still recruiting. So we have 36 patients with a phenotype of pure autonomic failure, and there have been many studies worldwide, so I don't have the time to mention that, but our particular interest was looking at how the accumulation of the alpha-synuclein differentiate our patient with a pure autonomic failure to the one that they phenoconvert to a different pathology. And we did describe, so the manuscript is in preparation, then the score. So the score means in how many structure we are able to document the alpha-synuclein in the skin. So pilomotor muscle, blood vessel, and the sweat gland is higher in patient with a pure autonomic failure compared to patients with multiple system atrophy. So then an additional study, which is again is ongoing, that was just a proof of concept, and now we are going on a larger cohort with many collaborators, actually UCLH and here Queen Square. It's very common for an autonomic neurologist actually see patients with a pure autonomic failure with a mild neurological feature. So a little bit of tremor, a little bit of bradycardia, then it doesn't allow you to neither make a diagnosis of multiple system atrophy, neither of Parkinson's disease. So in patients with high suspicious of a different pathology, so we did recruit 10 patients, and for us, high suspicious was a severe bladder dysfunction in the very early stage, which you would not expect in a patient with a stable pure autonomic failure, mild Parkinsonian feature, performing in that scan, which actually was abnormal in 50% of these patients with pure autonomic failure. As I said, it was a proof of concept, so we are going on to the larger cohort. We have a target of 100 patients. So far, we have recruited 70. So we are not done, but almost there, and pressure will be on Dr. Chiaro to conclude this study. But you can see then, obviously, we have follow-up this patient longitudinally, 11 patients from the time of the DAT scan. Unfortunately, they did convert into multiple system atrophy, 11 to DLB or Parkinson's disease. 48, they are still with a phenotype of pure autonomic failure, 4 of them with an abnormal DAT scan. If we are to combine DAT scan and IBG, and this is also, I had the question for Professor Pavese this morning, I believe that actually all these biomarkers is really, it depends in which phase of the disorder we are using them, yeah? So in this patient with the pure autonomic failure, combining that scan, MIBG, and the catecholamines, so you can see then 3 patients, they did convert into multiple system atrophy, 2 of them with the prodromal MSA, because the overall aim of our prospective study, as you can see at the end, is also validating the prodromal criteria. Patient with the abnormal DAT scan and abnormal MIBG, they were the one converting into DLB and Parkinson disease. Now, the reason why the question from this morning, because when I was at Mayo, we did a study with Kurt Kampinski and obviously with Philip Lowe, and in reality it depends when you use the MIBG, because in more advanced disorder, you can have a 30% that actually is positive in MSA. So to conclude on this initial part of the study, so we definitely have a wealth of knowledge now in several cohorts worldwide of biomarker that we can use to predict phenoconversion. I guess the challenge now is validating the prodromal, making sure that these biomarkers, they function very well in the prodromal phase, yeah? And this is where we are. So the reason why you see all these pictures, not because we party all the time, but because this is the results of a work of many people over time. Yeah, and this is where we are with our prospective study. So we did the phenotype with all the cardiovascular, pseudomotor, and catecholamines, 283 patients. Dr. Chiaro is trying to establish a yearly rate of phenoconversion. So what you see here. So the very dark green are patients, then they phenoconvert into multiple system atrophy. Majority of them, they would phenoconvert within 9 years. All the remaining, at least in our cohort, are patients with Lewy body pathology. Active on the study, there are 100 patients with a pure autonomic failure phenotype, 34, then they have converted to other pathology, and 61 patients, they died with a diagnosis of pure autonomic failure, 54 with the diagnosis of other alpha-synucleinopathy, and we have a 10% of loss at follow-up. So because of with the luxury of the plasma biobank established a very long time ago, we have all the sample for this patient, and in collaboration with the work that you have heard from Amanda this morning, we're going to measure several plasma biomarkers. And the remaining of the study, as I said, is validating the prodromal, assessing whether or not we have a diagnostic sensitivity of multi-dimension biomarkers. So we have heard that I don't think that we can address with one biomarker specificity, but also how we measure progression. So very likely we're going to end up with a cluster of biomarker, ideally some biomarker that actually can be used in many neuroscience centers. They don't require necessarily, obviously, an advanced autonomic center. And with the team also, we're going to obviously explore the seed amplification. Thank you for your attention to all our patients and many collaborators here.
So thank you very much. That's a great session. Can I ask all of the speakers, please, just to come and have a seat at the front? And I'm interested, so we got some hands up already for some interesting questions. So Chris Kobelecki there.
Thanks very much to everyone for those talks. I'd like to ask a question to Dr. Go, please. So the Innsbruck group last year published a prognostic nomogram for MSA patients, which identified several features, but it wasn't pathologically validated. Have you got any sort of comments about how that compares to what you found, or have you got any plans to try and produce some form of prediction device?
Well, I think a prediction device would be really good, and I have been looking at it. I think the issue that I have with the data that I have is that the data is potentially a bit messier than I would like in terms of using it to create a tool, if that makes sense.
Thank you.
But yeah, it is something that I've looked at before and have considered. But yeah, at present, I think the data that we have is just not quite as clean as we would like it to be.
Thank you. Yeah, please do.
Thank you, Boyd. Gosh, so I could probably ask questions to everybody, but I thought I'd pick on Wendi. Wendi, I was intrigued with the there were two things. One was a comment, which was, I guess the person with FND doesn't set out to look like MSA. It's the clinician's interpretation of their signs that suggests that they might have MSA, which I think you did make. But I was just going to say, this very slow, effortful movement of, when you ask them to do a finger tap and it's really painfully slow and looks effortful, it reminded me of the PSP sign of someone that's slow and fast from the start. And I wondered if that would be another positive sign for FND, or whether there's other pathology that would mimic that.
Well, with a lot of these functional signs, you've got to be careful about taking one in isolation. So, for example, this, walking on ice gait, it's just a cautious gait, which you see in lots of things. And particularly when you think about how common it is to have structural and functional things together, you just got to be a bit careful. Yeah.
Can I ask a question to the panel?
Yeah.
I think I want to comment on two things about with regard to effortful. That's been written up already. So this extra effort and attention to the effort as well, which is a clue. But I wanted to make a point. Sorry, these are the people with functional FND. But I just want to I was talking to Wendi a bit earlier, but there's a lot of literature coming out that FND symptoms are common in people, if particularly above the age of 50, those who are going on to go and develop something like Parkinson's disease or even frontotemporal dementia or other dementing conditions. Almost 20% of them will go on to do that. I don't think I've seen anyone with MSA, and I think you mentioned that, but it's an important thing to keep in mind that at the moment what you see is functional, but there may be an underlying organic condition coming on, maybe.
Yeah, sure.
So I've actually seen two people who've been diagnosed with FND who later came to my clinic with MSA, clear MSA. And I guess, again, it's our interpretation of what looks functional, which is our own biases, rather than their pathology that we can't see and we can't predict.
But you do get functional signs. You might get something that's purely functional, and then you get hidden structural signs, or they're not there yet. So I don't think that's a misdiagnosis.
It's not a misdiagnosis. It's like pseudo seizures in people with real seizures. So you don't call the pseudo seizure, know that this is also part of your epilepsy. The same thing applies here.
Yeah.
I have two questions for Yen. I'm gonna copy, Kailash on that. No, Yen, 2 points. So one about the survival. Then you have seen a different survival in the cohort. I guess the problem, as you said, obviously heterogeneous in the way that the study is designed, but also how you define onset. How you define onset. Because in some of the cohort, the onset was not defined by motor onset. It's an autonomic onset, yeah? So obviously, when you include the autonomic onset, you will have a patient that by the time they get to your clinic, they have 6 years of history. Yeah.
So our cohort was defined by autonomic onset as well.
Okay. And despite the autonomic, so the first symptoms, you have less in terms of disease duration, the survival, basically. You have a shorter survival in your cohort compared to...
Yeah, so with our cohort, the way the onset symptoms were defined was actually based on the other papers. So when we first, especially for the retrospective case review, the way I defined how to find out where the onset was by going back to those other papers and using their onset criteria. So on paper at least, the onset criteria is exactly the same. However, I also appreciate that sometimes on paper it can look the same, but in practice, the way it's collected, there could be differences. It's hard to know, but on paper it should be the same.
Okay. And the predictor of survival, having an early, so an autonomic onset doesn't predict survival, is it right? Okay. No, but in the other cohort is the same again. So it's not having an onset of autonomic, having an early severe autonomic failure. Yeah. Yeah. So I guess you have very similar data, which is what you want to have. Yeah. So something then So
yes, Claudio.
Yeah, I was very interested on the PF study. As you know, with Wolfgang, we did some years ago testing the CSF of people, and we correctly predicted the MSA and PD, but it was a very small number of subjects. So I think this is a very interesting study. I would like to really be able to do more. Of course, it's difficult with the CSF only, right? So as soon as we have the blood. The question I have is, why these people do not progress? I mean, why, have you analyzed the brain of any of these people that die without progressing? And is there synuclein, and how much, etc.?
So hopefully we will get more of, so in the prospective prodromal study, the aim is obviously, it's a difficult discussion, but it does happen in clinic all the time, yeah? So hoping then, the people, they will be willing to donate the brain, because at the end of the day, this is what it is. So there are only few case reports, yeah? So one described actually by Horatio, there is another, other 3 case report from the Bologna group and 1 from Goldstein. And patient, even without a clinical correlate, so in life, no neurological feature, at least what obviously we can read from the paper, they had Lewy body in central structure. So that is true, but it's also true that another case report, actually, a pure autonomic failure without any involvement centrally. So I suppose the big question is, which we don't know the answer, actually, so whether some of these patients, if they live long enough, yeah, they will all phenoconvert to other alpha-synucleinopathy, or actually there are patients and there are some protective factor, then they avoid the widespread pathology. But I don't think we can answer that question today.
Veronica?
Yeah, I would just like to do a comment, not really a question. I'll bring it back to genetics, of course, this PAF. So there is at least one gene I know that does cause pure autonomic failure and is not central nervous system, is peripheral. So I think...
But that is a different, I know which one
yes,
you are referring to.
But I think
a completely different phenotype.
Yeah. But I think there will be maybe in this group, those who never phenoconvert and do not have pathology, maybe a rare, smaller number of patients who may have a completely different,
but the
maybe
phenotype
genetic.
you are referring to is obviously is a metabolic, is a mitochondrial disorder, yeah. And from an autonomic standpoint, they have a different phenotype, yeah. So they have a very, very low category, very patchy cardiovascular. So we have one patient that actually has been studied by Vanderbilt and by you also later on. And potentially we have other 2 patients, yeah. Then actually they were admitted, one of the sisters was admitted last week. So, timing perfect for that question. They have a different phenotype when it comes to cardiovascular, yeah? So it's patchy. Our patient with the pure autonomic failure, they have a widespread autonomic failure, yeah? So parasympathetic is not working, sympathetic is not working, low catecholamines level, and the progression of the disorder is different, yeah? So the patient then you have tested, significant progression over time, very early onset, remember he started at the age of 20, yeah?
So for the
but
purposes...
you're absolutely right, and I guess this is, we were discussing with Henry this morning, I guess it's worth it in this cohort, looking at, potential genetic risk.
So we're running into tea breaks. I'm just going to ask for 2 more questions. We have Nicola and then there's one behind.
I'm actually answering to Valeria's question.
Okay.
Very nice talk to all of you. Yeah, you're right. It can be altered at some point. So it can be useful at the very beginning. Not at the very beginning, because as you have seen, there is some overlap with even Parkinson can be completely normal. And I wouldn't do when the disease is very established, even because as you presented that. The ideal to do the cardiac scan is at the time when patients have had diagnosis of Parkinson's for a few years, and then you start seeing that they are going in direction that is not the one that you would expect for a normal Parkinson's. So if that is a little bit prolonged more than what you would expect, that at some point can be useful. So is that in that stage that you would do it?
Thank you. Do you have a question? Yeah.
It's Eduardo Fernando from the Queen's Square Brain Bank. I just wanted to comment on this case with sort of pure autonomic failure phenotype that go on to to have an autopsy at the end. I mean, there is very few cases published in the literature. Some of them even published before synuclein antibodies were available, so we don't know exactly the pathological details of some of them, but from our experience in the brain bank here, what we see is that a lot of these cases that are diagnosed as pure autonomic failure, they convert, and they can convert quite late, even decades after developing autonomic symptoms. And even we have a few cases who did not develop any overt Parkinsonism in life, but when they go to autopsy, they have features of MSA or Lewy body in central structures. And I don't know if Zana is here, but I think pathologically, there is a lot of controversy whether this pure autonomic failure is a pathological entity or not, or whether it's just a peripheral or prodromal presentation of a Lewy body disorder.
Great. So I'd like to thank all of our discussants today.
Okay, I think we'll start the last session. So this is the therapy session. We're going to hear two talks, sort of different angles of approaching neurodegenerative disease. One is symptomatic, and one is what we hope is a disease-modifying approach. The first one's symptomatic treatment, and we shouldn't. It's easy to dismiss symptomatic treatments as physicians and think, well, it's only symptom control and stuff. But if you actually you survey patients, actually, of course they want a cure. Nobody's going to say they don't want a cure or disease-modifying treatment, but they do want management of the symptoms in the here and now. So these are important treatments to improve the quality of, let's be honest, this is a pretty awful disease, we all know. And if we can improve even some of the blood pressure control or some of the bladder symptoms, we will make a difference to the lives of patients with this disease. So first up is Professor Iodice, works here, colleague for many years. I think I was on your appointments panel many years ago. And we're going to hear about ampreloxetine, and its symptomatic manifestations on blood pressure. Thank you.
Thank you. So back with me. So this is a longitudinal analysis of the ampreloxetine for the treatment of symptomatic neurogenic orthostatic hypotension in a subset of patients with multiple system atrophy. And as Nick was just introducing, the symptomatic neurogenic orthostatic hypotension has many actually autonomic dysfunction, are common in patients with multiple system atrophy, and they do affect significantly the quality of life. Common symptoms of the neurogenic OH: dizziness, problem with vision, weakness, fatigue, trouble concentrating, head and neck discomfort. And this is something that obviously you have experienced with all your patients in clinic. There is a strong unmet need when it comes to treatment of neurogenic OH. We have not much in reality in terms of pharmacological portfolio of compounds. And the common therapy are droxidopa, which, as you know, is not approved in the UK, midodrine and fluidocortisone. However, despite this medication, maybe we will have in UK also the use of octreotide, then we use quite a lot, but many of the patients, up to 2/3 of the patients, which is in a way is quite shocking, do not respond to this therapy. And current therapy do not preferentially improve symptoms of end-standing blood pressure, and current depressor agent require multiple daily dosing, then obviously affect compliance. So the ampreloxetine is one of the really, maybe the only compound when it comes to the neurogenic OH to have such a targeting mechanism. Yeah? So, and I will describe what I meant with that. Was discovered and developed by Theravance, and is a selective norepinephrine reuptake inhibitor. Is orally administered as a single tablet, 10 milligrams, once a day. And there was a phase 3 registrational trial is currently enrolling. For the one of you that you did attend this meeting last year, we were saying, finger crossed, we're going to be open with the trial very soon, and now we can say actually the trial is active, many sites worldwide, so it's a brilliant opportunity for our patients. And the entire program, so you can see there was a proof of concept with a phase 2, there was a phase 3, then actually was started, I'll give you just a little bit of more details later on, in patients with all range of alpha-synucleinopathy, and now we are on the Cypress study with one additional pivotal trial in patients with multiple system atrophy. So what we meant with the targeting, so precise medicine when it comes to the treatment, so this is a NET inhibitor. So in patient, at least in patient where we would expect a pretty much an intact post-ganglionic function, the patient, they are not denervated. So if we have a way to increase the noradrenaline in the synapse here, obviously we can increase blood pressure, improving symptoms of orthostatic intolerance. And this is exactly what ampreloxetine is supposed to do. In terms of the entire program, so the Sequoia and the Redwood trial, so they started in 2019, and that was an immense opportunity for us to understand more regarding the compound, but also which patient they were really responding. So at the start of the program, as I said, were all alpha-synucleinopathy, so we were recruiting patients with multiple system atrophy, Parkinson's disease, and patients with a pure autonomic failure. But the ampreloxetine in the initial analysis, it didn't meet the primary endpoint, but on a pre-specified criteria regarding the multiple system atrophy, definitely the ampreloxetine in the initial trial was actually was absolutely positive in patients with multiple system atrophy. And you see some evidence here. So what we meant with improvement? So this is the design of the study. So you see the Sequoia, the Redwood, and you will see later on then the Cypress is very similar. So you have a 4 weeks double-blind period with compound and ampreloxetine. Then the patients are in the open-label period. So these are the orthostatic hypotension symptoms score. So that improve when the patient is on the open label. And at the end of this phase, the patient, they go on to a randomized withdrawal period where you would expect then actually they get worse because obviously they can be on the placebo arm. And this is exactly what we did observe. So it's not just about one of the domain of the orthostatic hypotension scale, but actually you can see in several of the domain, patients with multiple system atrophy, they did improve with the ampreloxetine. So that for us is very encouraging and definitely the drive to try an additional trial. Then when it comes to the blood pressure, the ampreloxetine also does improve the standing supine, the standing blood pressure, and there is no signal, at least in the previous trial, obviously things then obviously we will need to be able to confirm regarding the supine hypertension. Because as we have seen this morning, we have a huge challenge in management of the neurogenic OH because we have to balance not just the drop in blood pressure, but this patient, the baroreflex is not working. So blood pressure drops, but blood pressure can be very high when they are lying down. In terms of tolerability and safety of the drug, the adverse event during the randomized control phases, you can see that the ampreloxetine was generally very well tolerated, and overall the adverse event profile was very similar between placebo and ampreloxetine. And here we go with the new trial. So now the Cypress study is ongoing. So there are several centers. So these are the design, as I did mention, is pretty much similar. There are less visits, pretty much similar to the previous design. So the patient, they will have 12 weeks instead of the 4 weeks, 12 weeks open label with only 10 milligrams tablets per day. And there is an enrichment design criteria, so only the patient then they do respond, so where actually there is an improvement of the scale, they go on to the next phase. They are randomized in placebo or ampreloxetine, and then if they are responding, actually they are provided with the medication for 2 years, long-term extension trial, which is also another very important part of the design of the study. That means then we will have plenty of data also in terms of long-term effect of this medication in our patient with multiple system atrophy, which obviously something we were missing in previous trial like droxidopa and the very initial trial on midodrine. Who is eligible? Age more than 30 years, diagnosed with multiple system atrophy. Patient obviously have to have neurogenic orthostatic hypotension according to the consensus criteria, and needs to be symptomatic because that is one of the primary endpoint. So if you know any patient, then potentially can be eligible to the study. So there are 4 sites in UK where the patient, they can definitely be enrolled into the Cyprus, but also, and this is obviously we are all grateful to the sponsor, we are looking at also other options for patients and they are not served. So patients, they are not close to a site, then they can recruit them. We are thinking about other means to actually be as inclusive as possible. And the MSA Trust specialist nurse, then they will be in the room. They know exactly what I meant with that. So we're trying to support the patient and then actually they don't have an easy access to a site to be enrolled into the trial. So anyone that then does have a patient then is a possible candidate, I would be more than happy actually to have a discussion later. So thank you so much for your attention.
I just want to thank the speakers. I mean, it is exciting that there are, real prospects of trying to influence this disease, both symptomatically and from a disease causation point of view. For those who are younger in the audience, this might seem obvious, the sort of thing we might be doing, but it's not obvious for those who worked with MSA for many decades, as I have, and my colleague, Professor Bhatia, who's even older than me. He doesn't look it, but he is. And he's going to close the remarks.
Thanks, Nick. Definitely look younger than you. I know. So I have to do the closing remarks, and I'm probably wearing 2 hats here. So I work here and I work in MSA, but I'm also the chairman of the MSA Trust UK, taken over from Claire Fowler. And I must say this short period I've been with them has been really very rewarding for me to see the sort of work which goes on in the background. So firstly, of course, I want to thank all our speakers and all of you attendees, but I also want to thank the work which has gone on in the background from the MSA Trust UK, Karen Walker, our chief executive, our trustees, a number of the staff you might have met here, Eleanor, Emma Rushton, and so many behind the scenes. So they all have done a fantastic job to put this together. We had a symposium last year, and we were wondering, do we really need one this year? But Eureka was, yes, we need one. And as you can see, the number of attendees here, it has been very well received. So all good things probably come to an end, but I think this is actually a start, because the interest you all have shown here and what we've seen from all the presentations, I think that's a start to something new. I think finally we are getting around to something new. But in the MSA Trust and as a clinician, I think really this lies with the patient. And there are certain fundamental principles which we have in the MSA Trust. One of the first things I think which happens with patients who, when you first give them the diagnosis, the shock and the lack of information, and the question about where do we go from here. And as part of the trust, that's what we do. We provide information, support, education, and research. And I'm hoping that we'll add to this clinical trials and possible cures in the future. So with that sort of hope, I want to draw this meeting, which was really fantastic. I learned so much here, to a close. It's not over yet. So across the square, there is a very famous pub where if you want to meet Nick, you can find him in the evenings, generally. But half of Queen Square as well. So it's called Queen's Larder, and the reception, I believe, is over there. I also would be very grateful if you would fill in your feedback forms, and I think you're going to send them by email. On on Monday. So, and again, I think at the end I want to, give and felicitate our speakers and all of you for being here, and thank you for coming.